All right, good morning. Today is Tuesday, June 16th, and our goal, as you can see today, is to finally build that third step of our foundation by talking about tissues. So today is going to be a busy day that way, and then we'll hopefully in lab get a chance to finally start talking some histology and how to tackle that material. It's always scary when the schedule starts looking lighter and lighter because that means we're getting closer and closer to the exam, and that is indeed the case here. We only have 2 more lectures left after today, Thursday, and again, you have 2 assignments due on that, Unit 5 review, and then that biointeractive, where there's a handout that you're going to print out and then watch the video and answer the questions. And then on Friday, you have 3 assignments due. Your Unit 6 review is due. The epithelial tissue and connective tissue handouts, which hopefully you have today, so we can be looking at that and talking about what you're going to be responsible. That is going to expect to be completed with drawings, with labels. And again, I'm not grading you on your drawing skills, but you are going to be required to complete this. And again, the advantage of this is it helps you to familiarize yourself with this material, and it is your study guide for basically half of the exam. And then there's also a fun fingerprint activity. We are not doing the activity that's in your lab manual because obviously there's not enough of us together to do that. But go to the assignment, read the assignment. You're going to be taking a couple fingerprints of yourself or anybody else in the family that you want to. And then you are going to be basically doing 2 things. You're going to be identifying the general pattern of your fingerprint. And then there are the minutia of details, the tiny little details, or what are also known as Galton points. So those minutia and details, you're going to try to find, I think it's like 4 of those. And so you're going to use 2 fingerprints, and that should easily be able to find 4 of those. And so you will be using the chart that's on page 146 to identify that minutia of detail, those Galton points. For those. And then, like I said, as we've talked about, Monday is your lab and lecture exam, and it must be completed during the class time. All right, so with that in our pocket, unless there's any questions. Oh, actually, I lied. I do have some questions. So first I'll ask you, any questions before we get started? All right, excellent. The question I have for you, because we talked about it last time, but what I forgot to ask about was the results. Was there anybody who got all the questions correct on the practice lab exam? Is anybody who got a perfect 20 out of 20? Dead silence, okay. Is there anybody who got 0 out of 20 correct on them? All right, well then hopefully you fell somewhere in between those 2 things. What I wanted to remind you about that is to not put too much weight into how you did on that practice exam. It is a good gauge of how you're studying. If you did well, then that's good. That means that you've been studying the material in the appropriate way, and hopefully as long as you continue to do that, you will be successful on the real thing and also moving forward. If you weren't as successful on it as you wanted to be, then again, hopefully you didn't go home afterwards, or you were home yesterday. Hopefully after class, you didn't go back and flog yourself with a whip or something like that because of it. Again, it is a gauge to see how you're doing. What you should do is learn from it, appreciate that you have not been attacking the material in the right way. And then the good news is you have a week to change the way you're attacking this material to make sure that you're successful on the real thing. That was the real point of this, is both so that you could see what to expect on that lab exam, and also so that you could see how you were preparing so that you could change how you're preparing if you weren't as successful as you hoped to be. All right.
Question?
Yes.
For the next future exams, are we gonna keep doing the practice before the exam?
No. No,
Okay.
because again, the good news about this is once you've seen the first lab and the first lecture exam, you will now be familiar with the format of the lab and the lecture exam, so that you have you know what is expected of you, you know how to take it, you know what it's going to be like. And so the point of this was to familiarize yourself with it. Now, if there is time, and I will warn you, during summer, time is something we are more challenged with, but if there is time on the lecture before the exam, what I like to do at the end is to do a review. But a review is not where I sit there and tell you the things that I think are important. That is what I do every day, all day during lecture. What I see a review as is an opportunity for you to ask questions. So you can ask questions about things that you're not clear about, and we can work together to try to come up with what the answer is to that, or help you to understand those concepts better. So the reviews are question and answer. And so if we have the opportunity to do a review, then I expect you to ask questions. And if you guys don't ask questions, then I assume that you have mastered the material and I make the test harder. All right? So yeah, but no, this was just so that you could see what to expect. Most people have answered an essay question before. They know what a multiple choice question looks like. But for some people, this is the first time they've done a lab exam, and certainly the first time they've done a lab exam on an online A&P. So that was the point of that, is just to let you see what it would be like with one picture on there and one question on there and how you would answer that. But like I said, that was just about the format of it. All right?
Hi, Professor.
Yes.
For the exam, the leg exam will go first, right? You go first.
No, again, so again, if we were taking it in the classroom, because the lab exams are timed exams, usually we do the lab exam first because we all start at the same time and all end at the same time, and then we do the lecture exam. But the one thing that you do have the option of doing with this online format is you can actually take either exam in either order. So if you want to take the lecture exam first because you're more comfortable with that, or because you're worried it'll take more time and you want to see how long it takes you to take that, and then that you need to determine how long of a break you get after that for that, you can take them in either order. You can take the lab exam or the lecture exam first in either order that you want to take them. You just have to complete them both within the classroom.
Okay, so after we finish the leg exam, we can take a break, right? We
Yes.
can take a break and then go to the lecture exam.
True, absolutely. Or you could do it the other way around as well, because the lecture exam is longer. If you want to take that one first to see how long it takes you, and then you can, because like I said, I give you 2.5 hours, but if you're done in an hour and a half, well, then you have a lot more time before you have to take the lecture, before you have to take the lab exam. So again, you can take them in any order. You can take them however you want. You can take a break before or after, however, or in between, I mean, but they just both have to be completed within the class time.
Okay.
Yep. And in fact, I encourage you to take a break in between them, right? Because again, you need to calm down a little bit after one exam before you start the next one. So yeah, I don't encourage you going back to back with them. Take a break in between. So
when we take a break, we can go to the bathroom, right?
Yeah, exactly. If you're
leave the room, leave the.
Yeah, you're only being recorded during the exam. Once you end the exam, then, yeah, you're in your house and you can do whatever you want. Dance around the bedroom naked if you like. I don't care. Just don't do it on camera. All right?
Okay, got it. Thank you.
Excellent. All right, any other questions? All right. So we left off last time and we talked about our cell junctions, and we were getting ready to talk about our epithelial tissues. Now, epithelial tissues and one of the keys to our epithelial tissues, and where's my annotation? There we go, is that they are a cell-dense tissue type. What that means, kind of like the illustration that you see there in front of you, they are a bunch of cells packed very tightly, very closely together, right? Because of this, they typically have lots of cell junctions because they are so many cells packed very tight and closely together. And remember, as we also talked about, that is a good thing because our epithelial cells, and hold on, I have too much fun stuff up here on the screen. So let me move that. Let me move that. As we talked about, epithelial tissues are superficial. I think I can squeeze it in here. They're superficial. They're lining the surfaces inside and outside. So part of that, as we talked about, is needing to form a barrier. So when we talk about the anatomy of all epithelial tissues, and that's really what we're talking about here, all epithelial tissues and their characteristics, they are superficial, they're very cell dense, they have lots of cell junctions. When we identify them, they are cells tightly packed together and they're arranged in layers or sheets. To identify an epithelial tissue, There are basically two characteristics we need to take into consideration. The first is the shape of the cells. And as it turns out, there are three main shapes. Those three main shapes are first squamous, cuboidal, and columnar. Now, what do you think, let's start easy. What do you think the shape of a columnar cell is?
Column.
Yeah, column, kind of like the ones in the pictures there. They're tall, narrow cells in their setup. What do you think a cuboidal shape is?
Cube.
Squared.
Yeah, square, exactly. They're short, a block or a square shape cells. And so then that leaves us with squamous. What is squamous?
Square.
Sort of, but cuboid is square. We got to be careful with that. They can be squarish, but what does squamous actually mean?
He's nervous, squeamish.
Unshaped.
Actually, what it means is squashed, right? It comes from the same root of the word squashed. It's flat. Basically, what a squamous cell is a broad, flat shaped cell. So it's a broad, flat shaped cell. So we have these three main shapes, right? So we have, how are we going to do this? Let's cheat and use my drawing. So a squamous cell would be a broad, flat, oh, here, let's undo that. Let's do this instead. So if you think about it, it is a broad, flat cell that is very thin. Right? So it is a squamous cell, broad, thin, flat. In fact, sometimes these cells, if we looked at it from the edge, are so thin that their nucleus actually bulges out of them, kind of like a fried egg. Then, of course, cuboidal cells are going to be short, blocky cells, cube shape. And then our columnar cells are tall, thin, narrow cells that all sit side by side, like we see here, very uniform in their shape and orientation as well. So we have those three main shapes that we use for those. And now that I've drawn that, I have to erase all of that. The second characteristic we use to identify an epithelial tissue is we identify it by the number of layers. Right. When we identify epithelial tissues by the number of layers, it can either have one layer or it can have two or more layers. So if an epithelial tissue is made up of just a single layer of cells, we call it simple. If it has two or more layers, we call it stratified. Right? And so with that, you can just mix and match. So if I had a tissue, and now I will cheat and use my box. If I had a tissue that was comprised of a single layer of very flat cells, how would I identify that tissue?
Simple.
Okay.
Squamous.
Simple squamous. Excellent. It would be a simple squamous epithelial tissue. If conversely, I had a tissue that was made up of two layers of block-shaped cells, what would you call this tissue?
Stratified. Cuboidal. Epithelial.
Stratified cuboidal. There you go. Exactly. So you mix and match the shape of the cell, the number of layers, and you get the name of an epithelial tissue, like simple squamous, or stratified columnar, or something along those lines. You mix and match and put them together, and you get a epithelial tissue type. All right. So when we talk about them, they're arranged in sheets. They have 3 shapes, and they're either going to be simple or they're going to be stratified. All right. Questions on that? All right. Let's talk about the next characteristic of our epithelial tissues. Epithelial tissues, and again, here is the magic word that we are going to use. Epithelial tissues are polar. Now, up to this point in time, when we've talked about polar, we've been talking about ions, right? Things that are positively and negatively charged. That's not exactly what we mean here. An epithelial tissue doesn't have a partial or negative charge to it inherently within it that way. But what I mean is the same way that a magnet or even this pencil is polar, right? This pencil is polar. When I'm writing on my exam, does it matter which end of this pencil I use to write on the exam?
Yeah.
Yeah, as I say, it's not a trick question, right? I need to use this end. If I try to fill out an exam with this end, that's not going to be very successful. What we mean by polar is like the Earth. The Earth has a North Pole and a South Pole. A magnet has a North Pole and a South Pole, and they function differently. This pen has one end and another end, and those 2 sides are different from each other. So using the pencil this way and using the pencil this way is a very different function. Well, it's the same thing of this epithelial tissue. Look at the example of the picture that we have here. This particular picture tends to have these extensions on their outer surface. Any idea what these extensions might be? These really long extensions right here?
Cilia.
Yeah, you aren't sure? It says cilia right there on the label. Excellent. So if I were to take this cell and rotate it upside down inside the tissue so that the cilia were pointing down instead of up, would that change the function of that cell?
Yes.
Yeah, absolutely. And so that's what we talk about. When we talk about being polar, we talk about having 2 different surfaces, a top surface and a bottom surface. And again, because we're dealing with anatomists, anytime we talk about things, we can't use terms as simple as top and bottom. So we have to use appropriate anatomical terms. And the appropriate anatomical term for the top is apical. So we have an apical surface, which is the top of the epithelial tissue, and we have the basal surface, which is the bottom. All right? Now, there are 2 special things about epithelial tissues, and I'm going to go ahead and draw one again here. Let's draw this one so we can see. I'll do that. And then I will do. So what type of tissue am I drawing here?
Columnar.
Identify the tissue type.
Simple columnar.
Simple columnar. Excellent. And as we talked about, it has an apical surface and it has a basal surface. But there are a couple other characteristics about this we need to understand as well. Epithelial tissues are superficial. They're on top. So what is above an epithelial tissue?
The derm. I lost it.
All right. What is above an epithelial tissue? Do you have an epithelial tissue on your skin?
The outside surface.
Well, but okay. Do you have it's the surface? So this outer surface of our skin is an epithelial tissue, correct? We comfortable with that idea?
Yeah.
What's above it?
Just air, the outside environment.
Yeah, nothing. Space. Since epithelial tissues are the tops of things, then what is above them is space. So on the apical surface, there is always a space above it. Right?
Is there?
Yeah.
Is there dead layer above the epithelial tissue?
No, the dead layer is part of the epithelial tissue. As we'll see when we talk about the skin, you know, on Thursday and Friday. Yeah, the top, yes, you are absolutely correct. The top layers of cells of your skin are dead, but they're still considered part of the tissue. And because they're still part of the tissue, their apical surface is the top, and there's nothing above it. So there is always an apical space. And remember, these line surfaces inside and outside. I can take an epithelial tissue that is flat, that has a space above it, but I could then roll it up into a tube. If I roll it up into a tube, notice I still have that apical surface in there, and it still has a space above it. And when we enclose that space, when we have a lumen of an enclosed space like that, we call it a lumen. So let's cheat and draw this. Here is my hollow tube, like a blood vessel, like my stomach, like my digestive structure, or something along those lines. The hollow space inside of this is the lumen. So there's always an apical space, and sometimes that apical space is enclosed. And if that apical space is enclosed, we call it a lumen, right? So all lumens are apical spaces, but not all apical spaces are lumens, right? This is an apical space, but it's clearly not a lumen, right? But any lumen is an apical space. It always is above the apical surface. Now, if epithelial tissues are the top tissues, then that means that there is definitely going to be some kind of stuff underneath them. And when we have that stuff underneath it, and let's go ahead and put some stuff here underneath, we need to hold our epithelial tissue to the stuff, right? So what we need to do is we need to take a little bit of glue, and we need to put a glue on that basal surface so it will attach the epithelial tissue to whatever is underneath it. Now, because Mother's Day was right around the corner, you bought mom a really nice vase, and then you were roughhousing inside the house and knocked the vase over and broke it, you then use your super glue to glue it back together. And if you do a good job gluing it back together, do you see the glue?
No.
No, you don't see the glue. And that's the thing with this glue here. When we look at it under the microscope, with one exception we will get to a little bit later, we are never going to see the glue. We just know the glue is there. So what's going to happen is we are going to first point out the glue on this illustration because it's convenient because it's there. This is the glue right here. And this glue is what is known as the basement membrane. Or, because as we've mentioned, anatomists love to name everything, it is also referred to as the basal lamina. Basal means bottom, lamina means layer. This basement membrane is basically an extracellular matrix. Right? It's a substance that is outside of the cell, and basically it is the glue that holds the epithelial tissue to whatever is underneath it. All right? And when we actually look at some epithelial tissues, we will see that and talk about that. Now, yes.
So I'm looking at the picture, right? And we have like the basal lamina, which is more in the back towards the corner. And then we have the lateral surfaces. They're pointing, one of the basolateral is pointing at the same space as the basal lamina.
Okay, so there's two things I would say about that. Oh, not sure what's going on there. Okay, so you are correct, but here, let's be more specific. Let me do this. This basal lamina is referring to this entire blue substance that is on the bottom, which is, as I mentioned, the basement membrane or the basal lamina, right? Two names for the exact same thing, okay? What this is talking about is it is reminding you, as I mentioned, that there is a polarity to this tissue. we have the apical surface, we have the basal surface, and if you think about it, in between the cells, we have the lateral surfaces of these cells where they attach to others. Notice our illustration has some desmosomes here that are linking these cells together. And so what influences and affects the cell at the top and what can influence it from the bottom or the sides is going to be different. So what they're actually pointing to here are the basolateral surfaces, the basal and the lateral surface together called the basolateral surface. But you are right, on the exam, you guys can see my highlighter, right?
Yeah.
I could have this pointed right there on the exam, and I can actually ask two questions. Question one is I could ask you to identify the surface. And if I ask you to identify the surface, what is your answer to that? Anyone can answer.
Basal?
Basal surface, excellent. I could also leave my pointer there and ask you, and again, this will be exactly how I will word it, because remember, on a real tissue, you're not going to see it. What I would ask is, identify the extracellular material you would find in this location. And what's the extracellular material you would find in this location?
Basement membrane?
The basement membrane, or you could say
Basal lamina.
Yeah, there you go, perfect. Okay, so again, just like we talked about with the cell, that you can point at things, and again, keywords for key things is how we're going to be able to recognize and identify them.
So the name
Could
of the
you,
material.
Yeah, go ahead.
Yeah, the
You can-
material is basal lamina, correct? The material.
Yes, or basement membrane. That is correct.
Okay.
And then Nicole, were you asking a question as well?
Yeah, it was the same question. Thank you.
Okay. Yes. Yeah. So again, I wrote extracellular matrix, but again, let's do this instead. So the term I will actually use will be extracellular material. So if I want basement membrane, I will say identify the extracellular material, and let's go ahead and write it out. Found in this location. When we actually look at this, and let's actually cheat and quickly look at this. We'll come back to this in a second, but let's do this. Perfect. We're gonna, like I said, do a lot of histology today, but this is an okay starting point. Anybody have any idea what tissue type this might be?
Simple columnar.
Exactly. This is a simple columnar epithelial tissue. Identify the surface. Anyone? Nope, someone's chatting it.
It's not cilia,
That is
right?
correct. It is indeed the apical surface, right? Identify the space.
Is that the lumen? Or?
Lumen would be acceptable. What else could we call it? Apical space or free space. It's the free space or apical space. Excellent. Identify the surface.
Basal.
Basal surface. Excellent. Identify the extracellular material you would find in this location.
Basal membrane?
Not basal membrane. Basal lamina or basement membrane. Don't mix the two up. Okay, basal lamina, basement membrane, those are the two correct answers. Again, notice if the cell does it right, we don't see the glue, but we know this is where the glue is going to be. Okay, so like I said, we'll come back and we're going to do some histology together to help you guys get comfortable and more familiar with that. But we'll save that for later. All right, so let's go back to sharing this. Excellent. So that can go there. This can go there. That can go there. And I need print that again. Perfect. All right, excellent. All right, so we have that polarity to these epithelial tissues where they have a top with the free space above it, and we have the bottom with the basal lamina or the basement membrane on it. As I mentioned, epithelial tissues are very cell dense. They're tightly packed with cells. And so what that means is there isn't a lot of room for other things. One thing in particular there isn't a lot of room for is this right here. When I say all epithelial tissues are avascular, what does that mean? What does it mean to be avascular? Anyone? Means
Lack
that,
of blood.
yeah, exactly. It means that they do not have a direct blood supply to them. Again, this isn't something new to you. That's not something that you weren't aware of, because if you get an itch on your arm, what do? You scratch it. And while you're scratching it, you're definitely peeling off plenty of layers of cells, but do I bleed from doing that?
No.
No, right? Because it is avascular, right? If I want to bleed, I need to dig a lot deeper beneath that epithelial tissue down into the connective tissue underneath. So they are avascular. Now, again, it's important to realize that while they're avascular, these are not dead cells, right? In fact, they are dead tissues, right? We know there are some dead cells at the top of our skin, but there's plenty of living cells there as well. And again, it also, they're not unfunctional. While there are not blood vessels in our epithelial tissue, there are a large number of nerves. In fact, we receive a massive amount of information, like a fire hose worth of information is coming into you from your epithelial tissues. Some of it is as simple as when you drink that hot coffee or you eat that spicy food, but also the feel of your shirt on your shoulders, your chair on your back and your legs, your socks on your feet. Right? These are information that's coming into you constantly, but until someone like me reminds you of the feel of your chair on the back, or your socks on your feet, you're not thinking about it until then I make you aware of it again. So we're receiving massive amounts of information from our epithelial tissues. As I mentioned, epithelial tissues, because they're forming the boundaries between us and the outside world, have many cell junctions, and they are highly mitotic. Right? We are producing millions of new skin cells every single day. About every 4 weeks, you have, I'll answer that question in just a second. Every 4 weeks, you have a completely new epidermis on the surface of your skin. But even that's not that impressive. In your digestive system, in your small intestine, every 3 days, you are replacing those epithelial cells inside of your small intestine. So these cells are highly mitotic, rapidly dividing. And to answer your question, Madison, innervated means to have nerves in them, where we perceive information from them, that tactile sensation, that sensory information, pain, temperature, itch, pressure, touch, all of those things we get from our epithelial tissues.
Off-topic question. Do we replace these cells in our heart, or is it?
So yes, there is an epithelial tissue that lines the inside of the heart, and those cells can be regenerated and replaced. When we talk about the heart cells that are not able to regenerate, we're talking about the cardiac muscle.
Okay.
So it's the cardiac muscle that doesn't. But there is an epithelial tissue that lines the inner surface of the heart, and those epithelial tissues are mitotic and can divide.
Is there the same for the brain and for the spinal cord?
No. There are epithelial tissues on the surface of them, right? But not that. And again, we'll get to this when we get to the nervous system, but you think about a lot of tumors that occur in the brain are not actually of the nervous tissue, but in fact of the meninges. So you get like a meningioma or something along those lines, because it is the surrounding cells, the protective cells, that are able to divide, and not the nervous tissue itself.
Okay.
Yep. All righty. So these are our characteristics of all epithelial tissues, right? And again, your book's got a nice table that talks about these characteristics of all the epithelial tissues. But while these are characteristics of all the epithelial tissues, there are a lot of types of epithelial tissues, right? In fact, we can probably do the math and figure this out. How many different shapes of cells are there?
3.
3.
3. There were 3. Excellent. And how many different numbers of layers do we have to worry about?
2.
Right, either simple or stratified. So 3 times 2, how many
6.
specific types of epithelial tissues are there?
6.
Exactly. 8 specific types. Wait, what? As I told you, anatomists hate you. Absolutely. Based on the rules as we have described it, there should be 6 epithelial tissues, but there are actually 8 specific types of epithelial tissues because anatomists hate you. They make up rules and then they change the rules on us. They like to throw curveballs. So let's start with the easy ones first. Our simple epithelial tissues or epithelia, and there are 4 specific types. 3 of them we could figure out. Simple squamous, simple cuboidal, and simple columnar. Here we see the simple squamous. Notice if you think about it, this simple squamous is the thinnest possible tissue type that we could make for an epithelial tissue. It is just a single layer of flat cells. So would this be something that we would want, for instance, on the outer surface of the body, providing protection for us? No. And even a single layer of columnar cells isn't going to provide a lot of protection. So these simple epithelial tissues, because they are thinner in nature, are better suited for absorption and secretion. And so those are where we're going to see them for their functions, right?
Can you give us an example of where each of those tissues would be?
I am going to do exactly that, absolutely.
Thank you.
So let's talk about simple squamous first. Simple squamous is thin, so it forms a thinnest barrier. So again, this is going to be useful for internal barriers. Oops, wait, hold on, back. So when we just want to separate one thing from something else inside the body, that can be useful that way. And we'll see some examples. In fact, we're going to look at the very first tissue slide we look at in the kidney. We're going to see a simple squamous there. But I think where people think about the simple squamous the most is in the lungs. How vitally important is it for you to get oxygen into your body? Is that relatively important? Right?
Yeah.
If someone were to put a plastic bag over your head for a prolonged period of time, that might be an issue for you? Yeah, absolutely, right? That would definitely be an issue. Don't do that. All right, it is vitally important we get oxygen into our body. It is equally important that we get carbon dioxide out of our body. And yet, I will tell you that gas exchange, the movement of oxygen into our body and CO2 out of our body, is a completely passive process. Right? Remind me again what it means to be passive.
Doesn't use ATP.
Does not use ATP, exactly. So as vitally important as it is to get oxygen in and CO2 out, we don't actually use any ATP in that process. Instead, we make use of our diffusion rules. Your lungs folded up inside of your chest have the surface area of pretty much a tennis court. So you have a tennis court's worth of surface area inside your lungs. And on top of that also, we have a simple squamous epithelial tissue as the membrane. So that what we're able to do with that is we are able to have the shortest distance that the gases need to be able to travel for us to be able to get the oxygen in and the CO2 out. So therefore, gas exchange in the lungs is one of the places where we'll find the simple squamous, but it also, because it is thin, is useful for just forming internal barriers as well. Simple cuboidal is a very important tissue that is used a lot for making the tubes And again, I'm going to put this in quote, of the bodies. And by tubes in this case, what I'm really talking about are glands and ducts. When we're making our glands and ducts, they're going to be lined with cuboidal tissues. In our kidney, where we're processing our filtrate, where we're making cleaning our blood and producing urine as a result of that, you're going to find simple cuboidal epithelial tissue. So it's found in a lot of different places, like the glands, like the ducts, like the kidneys, and other places like that as well. Simple columnar epithelial tissue is primarily found in the digestive system. However, we can be more specific. If you think about it, it's not in the openings, right? In our mouth where we have that hot coffee that we're pouring in, or that hot pocket you just pulled out of the microwave that is molten lava hot. We wouldn't want a tissue like that in our mouth or even our esophagus to be able to protect us from that. But from the distal third of the esophagus all the way to the rectum. So not the anus, the exit point of the digestive system, but basically from the esophagus, through the stomach, through the small intestine, through the large intestine, all the way to the rectum. That entire surface is lined with a simple columnar epithelial tissue. And notice something about this picture. While it's not required, notice many of these have those microvilli. Remind me again, were microvilli the one that moved or the ones that didn't move?
The ones that didn't move.
Right, they're the ones that are there to increase the surface area. And again, if you think about it, your small intestine, your large intestine, these are areas where we're absorbing all that massive amounts of nutrients, that cheeseburger you had for breakfast. We're absorbing all those things there. So frequently, we will see microvilli on those simple columnar cells. All right, so these are the three obvious simple epithelial tissues and where we would find them in the body and what their general functions are. Questions on these? All right, so then let's talk about the curveball. The curveball that our anatomists give us is this: the ciliated pseudostratified columnar epithelial tissue. Now, every single section, every single portion of this class, there is going to be an alphabet soup term which you can be guaranteed that you're going to have to spell at some point or another. And in this case, this is the one for this section. Ciliated pseudostratified columnar epithelial tissue. All right. And again, notice there are three words in that. The order isn't as important, but you must have all three words there. So let's talk about all three words, starting first with the cilia. Right? Ciliated is simple. It means it has cilia, and those cilia are always going to be on the apical surface. And remember, cilia are the ones that are motile. These are the ones that move in a wave-like fashion, right? Again, not to move the cells, but to move substances across their surfaces. So that's the easy one. So let's talk about the other two terms. Starting first with pseudo-stratified. What does pseudo mean?
Semi or?
No, it's even less than semi. What does pseudo mean?
It's like a fake version?
There you go, fake or false, right? Chemistry is a pseudoscience, right? So pseudo means fake or false, right? Well, in this case, this is a pseudo-stratified. What that means is as you look at it, notice, and let's go back actually, hold on, I'm gonna clear the pictures and let's go back for a second. Notice with our simple columnar, all the cells are fairly uniform in shape. All of the nuclei kind of line up like ducks in a row. They're all very similar in their appearance. Notice here, whether we look at the illustration or the actual light microscopy of it, notice the nuclei are all over the place. This looks like it is made up of multiple layers of cells, but it turns out it actually isn't. Instead, what we have are irregular shaped columns that are pointing in all sorts of different directions. So some are shorter, some are longer, some are pointed one way, some are pointed the other way. So remember, when we look at this under the microscope, we are cutting a thin slice of a three-dimensional structure. And when you do that, you cut through all these different long column-shaped cells at different angles. And when you cut through them at different angles, it looks like there are different layers. So it looks like it has different layers. It cheats you into thinking it is a stratified tissue. But there are a couple things that are going to help you to be able to realize that it isn't. Typically, with a stratified tissue, there will be nuclei near the apical surface. This one, notice there, all the nuclei are near the bottom. There aren't any real nuclei up here near the top. Right? And of course, the dead giveaway is the cilia. If you see cilia, then you know it's trying to trick you into thinking it is stratified when it really isn't. All right? So these are the keys you're going to be using to try to help to identify this. What I want you to do right now is to focus right here at this tissue. Because when you focus right here on this tissue and we move to our stratified tissues, when you look at the basal layers of our stratified tissues, the basal layers of this ciliated pseudostratified tissue, they're all going to look the same. One of the challenges with epithelial tissues is some of them look like some of the others, especially at the basal surface. So if you really want to know what an epithelial tissue is, you need to look at the apical surface. At the apical surface, we see there aren't any nuclei, and we see these long, extensive cilia. And so by looking at the top, that is truly what is going to tell us what this tissue is. All right? Now, as we mentioned, these cilia move substances, and the primary substance that they move is they move mucus. Oops, I spelled mucus right. There we go. It moves mucus. So this ciliated pseudostratified columnar epithelial tissue is primarily found in two locations. Remember, as we talked about when we talked about cilia originally, the nasal cavity. In your nasal cavity, you have this ciliated pseudostratified columnar, which is capturing the mucus and moving it to the back of throat so you can swallow or spit it out. The other place you find this is in your trachea. Your trachea has these cilia that take the mucus that is being produced in our airways and moves it up towards our larynx, towards our throat. So again, it can either be spit up or swallowed. It's actually called our mucus elevator. But remember, as we talked about, these cilia are sensitive. So let's say, oh, you're a chronic pack-and-a-half smoker a day, all right? What happens is constantly smoking those cigars or that cigarette or vaping damages those cilia. And so what happens is now the cilia aren't functional anymore. So now that mucus sits in your trachea. And if you're a chronic smoker, then how do you have to remove that mucus?
Coughing?
Yeah, that horrible smoker's hack. Because that's the only way to move that mucus out. One of the concerns if you're a serious smoker is that if you then get a cold and take a cough suppressant, right? You've taken that cough suppressant to stop the coughing, but the problem is your cilia aren't working and now your cough reflex isn't working, and all that mucus with all the dust and the debris and the toxins in it is going to drain down into your lungs, dramatically increasing your likelihood of COPD or lung cancer or even just pneumonia or other types of lung infections. So again, it is take-home message: don't smoke. But the point is, those are the two areas: the trachea and the nasal cavity is where you find this ciliated pseudostratified columnar epithelium.
So, is this ciliated tissue also have a relation with the alveoli in the lungs?
No, Because again, if you think about it, we wouldn't want these in the alveoli of the lungs because it'd be way too thick for us to do that rapid gas exchange. The alveoli in the lungs is where we find the simple squamous.
Okay.
So this is just in the trachea. And then what ends up happening is our epithelial tissues transition from the trachea to the alveoli. And there in the alveoli, we finally get the simple squamous for the rapid gas exchange.
Okay.
All right. Any questions on our simple epithelial tissues?
Yeah, I have one question. So this part right now, is this made out of just columnars, correct?
Yes, irregular shaped columnar cells, yes.
And they're considered simple, correct?
Yes, because it is one layer. Some are just shorter, some are longer, and they point at, weird directions. So it's still, so if we straighten them all out, what you would see is that there's just one layer of cells of different lengths, but all column shapes that form that, and then they have the cilia on top.
So what is the difference between the? Okay.
Professor.
Yes.
On the microscope, we can see the microvilli, right?
Yep, absolutely. That's what you see. This picture over here on the right is actually from a light microscope. Yes.
Okay.
So notice we see the nucleus. In fact, not only do we see the nucleus, but notice we also see the dark spots inside it. We see some nucleoli inside of these nuclei. And then we see the cilia on top.
We can see the microvilli.
Yes, you can see microvilli as well, but microvilli are typically shorter than cilia. In fact, now that we know, when I showed you this picture before, remember there was this darker spot up here on the top? Guess what that up there is? Identify the organelle.
Okay.
So identify the organelle. Anyone? Identify the organelle. Oops, didn't want to do that.
Is it cilia or microvilli?
Microvilli. Notice this
Sorry.
remembers the simple columnar. Simple columnar have microvilli on the top of them, whereas ciliated pseudostratified columnar have cilia on top of them. Okay, but like I said, we'll come back and do that later. Let's go back to the lecture. All right, any other questions on our simple epithelial tissues? All right, excellent. Then let's identify our stratified epithelial tissues. Again, we have, well, let's do this first. We know with a stratified epithelial tissue, we know it is 2 or more layers of cells. But what if I have a tissue that is made up of a single layer of flat cells on top of 3 layers of cube-shaped cells on top of 2 layers of columnar cells. What the heck kind of tissue would this be?
Adipose.
Combo plate? Anyone else? What kind of tissue would this be?
Do we say, like, we start with the top layer, squamous, and then we use that fake word, I forgot how do you say that, the previous picture?
The pseudo?
Yeah.
But remember, this one actually is made up of multiple layers, so we wouldn't necessarily use that. It's a little tricky, isn't it? Well, here's the answer. This particular tissue is a stratified squamous epithelial tissue. Because our rule for identifying stratified tissues is A, that it is 2 or more cell layers, And B, the second rule, is we identify it by whatever tissue or whatever cell type is on top. Or let's be more specific, is apical. Whatever is apical, that is how we identify this. So it could be 543 layers of cube-shaped cells and 1 layer of flat cells on top, and it's going to be a stratified squamous. We always identify it by what is on the apical surface. And let's be clear about that too. With 1, 2, 3, 4, 5, 6 layers of cells, how many apical surfaces does it have?
Only has one surface, right?
Yeah, exactly. So there's only one apical surface. And then, of course, above that apical surface is the apical space or the free space, right? And so then how many basal surfaces is it going to have?
1, 2.
No, just one. One basal surface, the bottom.
Wouldn't each, what would each section be identified as then?
Different layers. These are
okay,
all
so
different layers. Yeah, so there we don't have to worry about the name of whatever this is between the layers. Yep, nope, but just when we talk about surfaces, there's one top and one bottom. No matter how many cells are making it up, there is one top and one bottom. One apical surface with the free space, one basal surface, and again, remember the basal surface is where we find the basement membrane.
Oh, okay, so there's only one attic and one basement in the house.
Exactly. And only one basement membrane and only one free space. All right? So now that we understand that, we can take a look at the pretty picture from your textbook. And we can see here is a stratified squamous. Notice cuboidal and columnar cells down here near the bottom, but up at the top, it is flat. Here we have 2 layers of cube cells. That makes it easy, cuboidal. But notice here, 2 layers of cuboidal cells with 1 columnar on top. And again, it's what's apical that matters. All right? Now, on the exam, if I ask you to identify and describe the epithelial tissues, you need to be able to identify and describe all 8. However, if you look at your epithelial tissue handout, the study guide for the lab exam, do you see stratified cuboidal and stratified columnar on that list?
Yes.
Really? Show me. Show me where on this you see stratified cuboidal and stratified columnar.
No.
No, it's not there. Because these 2 tissues are very rare in the body. They are typically found in one place and one place only. They're typically found in very large glands. And that's it. They're just found in very large glands. That's really the only place where we find them. Okay? As such, I am not holding you responsible. You do not need to identify them histologically on the lab exam. All right? If I, in an essay question, ask you to identify and describe all of your epithelial tissues, then absolutely you have to talk about them. They're some of your epithelial tissues. But on the lab exam, none of the questions will the correct answer be stratified cuboidal or stratified columnar. You are not responsible for them histologically. Does that make sense? All right, that is good. However, if you also look at your epithelial tissue handout, what you'll notice is that there are actually 2 specific types of stratified squamous epithelial tissues. Now, I know all of you are safe and home and not in the lab, but I'm still going to ask you to do this as a thought experiment and not as a real experiment, all right? As a thought experiment, if you think about it, well, this first part you can do. Rub the outside of your cheek, all right? See what that feels like. Now, the other thing you could do, and again, I'm not encouraging you to do this as a thought experiment. You could rub the inside of your mouth, the cheek on the inside, right, with your finger. Now, if you were to actually do that, do those two things feel the same? Does the inside of your cheek and the outside of your cheek feel the same? No, but guess what? Both are stratified squamous epithelial tissues. They don't feel the same, but they're actually both stratified squamous epithelial tissues because you have two types. The difference between your cheek and the inside of your mouth is the inside of your mouth is wet and moist, and the outside of your skin is hard and dry. And the reason for that is these cells on your skin have a special protein inside of them. That special protein is called keratin. It hardens and dries your cells. And so this skin on this stratified squamous epithelial tissue on your cheek has keratin in it, and we refer to it as a keratinized stratified squamous epithelial tissue. And the inside of your mouth, those cells don't have keratin. So they're a non-keratinized stratified squamous epithelial tissue. So as we look at these things histologically, there's actually two different types of stratified squamous epithelial tissues we are going to identify. The keratinized stratified squamous and the non-keratinized stratified squamous. And again, the keratinized is found in the skin. That is the only place we find the keratinized. The non-keratinized, and again, if you look at this tissue, you can see it is comprised of a large number of cells packed together. It provides a lot of protection. And so that's really the key for all of our stratified. All of our stratified are going to provide protection, but especially these stratified squamouses. This non-keratinized stratified squamous basically protects the wet openings, the moist openings of the body. Like what? Give me an example of the locations then where you would find a non-keratinized stratified squamous epithelial tissue.
Esophagus?
Yep, mouth and esophagus, oral cavity, esophagus, absolutely. But remember, just the top 2/3 of the esophagus. The bottom third of the esophagus, remember, is going to be simple columnar. But absolutely, the top part, the larynx, the pharynx, I should say, the throat, your oral cavity. What are the other openings to the body that need to be protected from use and abuse?
What about like the eyeball? What does that have?
Yep. Over the surface of the eyeball, absolutely. You have a non-keratinized stratified squamous epithelial tissue over the sclera, over the white part, called your conjunctiva. Right? Again, most of you have your cameras off. You should be able to at least look and find the openings of your body. It's anatomy and physiology class. It's okay to say things like anus, things like urethra, things like vagina, right? All of those are openings to the outside world that need to be protected from the outside world. So all those openings, all those moist openings to the body are going to be lined with a non-keratinized stratified squamous epithelial tissue. All right? Like I said, you have that spicy food, you have that sharp tortilla tip, you have that spicy coffee, right? The urethra is an opening to the outside world. Right? The anus is an opening to the outside world. The vagina is an opening to the outside world, right? Reproduction, if done appropriately, should be vigorous. We need protection in there, right? So again, we want to protect ourselves from the outside world. So having lots of areas, lots of cells packed together provide that protection from friction, from, again, from chemical, from abrasion, from all those types of things. All right? Questions on that? All right, when we get to the histology, we will talk more about these tissues and identify the components of them that you need to know. Oh, but we forgot our curveball. our curveball for the stratified epithelial tissues, because again, there are 4 specific types. So I spelled types that was correctly. I get 1 letter just like you. Is this one, transitional epithelial tissue. Transitional, as the name indicates, means that it can change. And in fact, we see that from its shape, right? Again, this is one of those points in the class where we get to talk about an important life lesson. What makes the world go round? Come on, give me your answers. What makes the world go round? It's the most important force in the universe. I'll wait till at least someone gives me one answer. Gravity. Gravity's a pretty good one. That's not bad. What else?
Gas.
Gas. Sure, maybe. Right. I know. We'll see. You guys are a little bit more down to earth than I am. I usually think when I ask what makes the world go round, people give me answers like love. Right. Love makes the world go round. Money, especially nowadays. Right. Money seems to be what's driving the reopening of the world, even though coronavirus is still running rampant. Right, all of those things, your mom, all of those are great things. But what actually makes the world go round, the most important force in the universe is pressure. Pressure is what makes the world go round. And so a lot of the things that the body does is to deal with pressure. And this is a classic example of it, right? If I had a balloon, and this would have been a great time to have one of those, but I don't have one. If I had a balloon and just put a teeny bit of air in it and then let it go, what would happen?
It will just move a little bit and
yeah, it would just pretty much drop to the ground. But what if I filled that balloon up and let it go? What would happen?
It will travel
Around the room.
over there.
It would fly around the room. And what is the force that would cause it to fly around the room?
The air that you put in the balloon.
Right. And more importantly, the air stretched the elasticity of that balloon, and that balloon has elasticity and wants to recoil. It puts pressure on the air, and the more air inside of it, the more pressure it puts on it, and the greater the pressure, the faster the air is pushed out and the more it flies around the room. Right? Pressure is what makes the world go round. Well, notice this transitional epithelial tissue is located in your bladder. So there you are in your car, driving along the road, and you realize you have to pee. And the problem is you just passed the sign that said the next rest stop was 34 miles away. You have 2 choices. You can pull over to the side of the road and find a bush that you can pee behind, or you can keep driving for another half an hour till you finally get to that rest stop. As you continue to drive to that rest stop, your bladder continues to fill more and more and more and more. And when you finally get to the rest stop and start to release your urine, do you fly around the room like that balloon from all the pressure of that urine being released from your body?
No.
No, of course not. And the reason for that is this transitional epithelial tissue. Notice on the top picture here, you can see the bladder when it is empty, these cells are tall and columnar in shape. But what happens is this tissue has the ability to stretch, to expand, not with elasticity like that balloon, but by changing the shapes of the cells. And what this allows the cells to do is it allows us to expand the volume of the bladder without dramatically increasing the pressure. Now, is the pressure of the urine leaving your bladder when it's full versus when it's half full greater? Yes, but not the same way it is with the balloon. What this tissue, this transitional tissue is able to do is it is able to change its shape, and that dramatically decreases, it dramatically decreases the pressure that builds up in the bladder. In fact, a transitional epithelial tissue can conceivably be stretched out till it would be a single layer of flat cells. You're absolutely correct. The uterus would be a perfect location to find this tissue, because again, after all, you have to go from that single cell to a basketball inside of you, but in fact, it's not found there. It would be a really great place to have it, and it would make sense to do that. But it turns out this transitional epithelial tissue is in the urinary system only. So the four structures that we see here, the two ureters, which bring the urine from the kidneys to the bladder itself, and then the urethra, all the way to the end. Remember, the tip of the urethra is going to be that non-keratinized stratified squamous. But the urethra, the ureters, and the bladder, those four locations, this transitional epithelial tissue is only found in the urinary system. All right? Questions on that? All right, excellent. With that, we are done with our epithelial tissues, identifying the 8 specific types, what their anatomy is and what they're looking at. And again, we'll look at them histologically in just a second as well. But for right now, let's go ahead and take our first break. It is 9:20, so let's take a 15-minute break, come back at 9:35. All right, any questions on any of that? So we have talked about the specific type of epithelial tissues. We've talked about the characteristics that all epithelial tissues share. There's one other piece of information we need to know about epithelial tissues before we can start looking at them critically histologically, and that is to know that epithelial tissues are what are responsible for producing glands. So all glands are formed from epithelial tissues. All right, now one of the important things to remember is first what a definition of a gland is. A gland can be a single cell. There are many, many single-celled glands that are found within the human body. Or it can be a mass of cells that are specialized for secretion, producing a substance and releasing it from the cell. Now, typically in the body, there are 2 main types of glands, endocrine glands and exocrine glands. So one of the first things we need to be able to do is to define these different types of glands. Now, hold on, juggle some things around here. Excellent. The first thing we want to do is we want to define things, right? So for starters, exocrine, like most things, we can break them down into the root. What does exo mean?
Out of?
Outside, absolutely, it means outside. Oops, outside, I'm not Canadian. There we go. Outside, excellent. Outside. Anyone know what crine means?
Is that gland? Or?
Close. Yeah, it means to secrete. Excellent. Yeah, crine actually means to secrete. So literally, exocrine means to secrete outside of the body. Give me an example of an exocrine gland.
Like sweat?
Yeah, sweat
Maybe.
glands. Sweat glands would be a good example. Any others?
Tears?
Nasal drip.
Well, you are correct in that the mucus.
Oh, it's not a gland though.
Right, but you are right in that there are mucus glands in the nasal cavity that produce mucus, and that would be released outside of the body. Absolutely.
Lacrimal?
Lacrimal is a great one. Perfect. Right, your tears. What else? Give me at least one more.
Mammary? Ooh.
Oh, mammary is a great one. Perfect. Excellent. Notice what I didn't hear anybody say was salivary. Would salivary be an example of an exocrine gland?
Yes, because technically outside the body.
Right. Remember, I think we talked about that already, right? Remember the lumen of our digestive system. Right. We have that hollow tube that is our digestive system from mouth to anus. And remember, as we talked about, the lumen of that, as we mentioned, is the topological exterior of the body. And so, yes, absolutely. Any kind of salivary gland or other type of gland, the hydrochloric acid glands in the stomach, all those things, intestinal glands, all secrete into the digestive system, which is outside of the body. So absolutely, that is perfectly correct. Now, one of the keys to these exocrine glands, as we'll talk about, and so I need to cheat and erase this now, is that they secrete outside the body, and they do so with ducts. Basically, they have two parts. The two parts of the exocrine gland are the secretory part, which of course makes the secretion, but it also has ducts. Ducts are conduits, are passageways. They're like PVC pipe. Their job is to just carry substance from one place to another, right? You've got sprinklers out in your backyard, and the PVC pipe carries it from one location to another. But does the PVC pipe make the water or change the composition of the water or do anything else to it along its way?
No.
No, it's just a conduit or a passageway of getting there, and that's what the ducts are. So the ducts are just the passages, right? The conduits for the substance to be released from the body. All right, so that's what it is. Basically, it's able to carry these things and carry it outside of the body, and it does it via those ducts. So it has those two main components to it, and it secretes outside the body. Now, based on our definitions then over here, we know crine still means to secrete. So we still have that to secrete. And of course, what does endo mean?
Inside?
Within.
Within, absolutely. Endo means within or inside. So endocrine glands secrete within the body, right? And so that's the key. These are going to secrete their substance inside the body. And really, what they do is they release it into the humors of the body. What do I mean by the humors of the body? What are the humors? Are those the funny things?
No, that's the humerus.
Yeah. No, the humors of the body are basically the fluids of the body, right? And in this case, primarily, they release into the interstitial fluid. Of course, once it gets into the interstitial fluid, where does it go from that?
Out everywhere.
Right, and how does it get everywhere?
Through the blood system.
Bingo, absolutely. From the interstitial fluid, it basically goes into the blood. And once it gets into the blood, as you mentioned, it can go everywhere, pretty much having the potential to affect every cell in the body. Now, it doesn't mean it's going to affect every cell in the body, but the potential is there, right? So notice endocrine glands are ductless. I don't know if that's an actual word, but I'm using it as an actual word. Exocrines have ducts, release outside the body. Endocrine glands are ductless. They just release their substance into the interstitial fluid, which gets into the blood and carries it anywhere. And to finish off our definitions, what of course is it that those endocrine glands are producing and releasing that could affect every cell in the body?
Hormones.
Hormones, absolutely. These endocrine glands produce hormones. Exactly. All right. Now, endocrine glands, we will talk about, we need to know the difference of definitions of these, but obviously the endocrine glands we will talk about in much more detail when we get to the endocrine system in 431. So what we are going to be focusing on right now is primarily focusing on our exocrine glands. So our goal is going to be to ignore those endocrine glands for now and to focus on our exocrine glands. Now, when we identify exocrine glands, there we go. There is basically two ways we define them.
Cody, can you mute your mic, please?
Yeah, I've got it. Remember, the two ways that we can define them are going to be, like a lot of things we do in this class, structurally and functionally. And of course, as we know, structurally is basically how they're made. And functionally, how they are made. There we go, that way. How they're made. Whereas functionally is how they release their substance. All right, so again, most of the things we're going to talk about in this class, we're going to talk about functionally and structurally. So you are going to be responsible for knowing all of the structural classifications for exocrine glands. You're going to be responsible for knowing all of the functional classifications of exocrine glands, and those make excellent essay questions. However, from now till the end of 431, every single time we identify a gland, there is going to be 3 important things you're going to need to know about it. Those 3 things you must know are its structural classification, its functional classification, and the substance it produces. So we're going to need to know this 2 ways. You are, like I said, a possible essay question would be identify and describe all of the structural classifications of exocrine glands. A possible essay question would be identify and describe all the functional classifications of exocrine glands. But also, for instance, on a lab exam, I may show you a picture of a sebaceous gland, which happens to be one of the oil-producing glands of the skin. And 4 of the possible questions I could ask you is first, identify the gland, and you would say sebaceous gland. But I could also ask you its structural classification, and you're going to need to know that it's a simple branched alveolar gland. I could ask you the functional classification, and you're going to need to know that it uses the holocrine mode of secretion, and you're going to need to know the substance it produces, and you're going to say sebum, and the function of that substance as well. So we are going to learn this information now as a possible essay question, but every single gland you run into from now to the end of 431, you're going to be using this information. You need to know their structural classifications, their functional classifications, the substance they produce, and what that substance does. All right, so let's go ahead and clear all of that. Remind you of that with the pretty picture. There you go. We're going to classify glands structurally, functionally, and by the substance that they secrete. All right, let's start first with our structural classifications. Now, let's start first with the simplest definition of a structural classification. When we look at the structural classification of the gland, the very first question we have to ask is it unicellular or multicellular? Of course, if it is unicellular, what does that mean? It's not a trick question. What does it mean to be unicellular?
One type of cell?
One.
United.
It is a single cell gland. Bingo. That's it. A single cell that makes a secretion that releases it. Here's a pretty picture of one of the classic examples, one of the most common examples found in the body. And that right here is what is known as a goblet cell. Now, we haven't looked at any histology slides yet. We're going to do that at the end of this portion when I talk about the glands. But one of the things that you can see, this is, guess, any idea what tissue type this might be that we're looking at? Someone want to hazard a guess?
Basal stratus.
Yeah, in fact, it says it right here on it, right? It's a ciliated, pseudostratified, columnar epithelial tissue. Excellent, right? We can clearly see the cilia on top. Now, if we were to look at this tissue in your body, would it really be this bright, beautiful pink that we're seeing right here?
No.
No. Most of the time, you got to think about how we make these slides. The way we make these slides is we take a chunk of tissue out of an organism, and then we freeze it. Once we freeze it, we use an incredibly sharp blade to take off just like 30 micron slices of this tissue. We then take that really thin piece, we mix it with all these different chemicals and stains to increase the contrast of the things that we can see there. Things like color that we see here are typically not true to the way it looks in the body. And we have a fancy term we use for that. Something that we see on the tissue that isn't the way it really is in the body is something we call an artifact. Sometimes that artifact could be damage, because when you're freezing the tissue and slicing the tissue and putting it with all these chemicals, the cells or the tissues could be damaged. Sometimes the damage is put there on purpose. Maybe you're using a piece of slide that you want to orient yourself so you know where you are. So one of the things they'll do is they'll take a needle and poke a hole in the tissue so that it's easy to line up all the slices, because you have that hole that you can use to line those up. And color is another example of an artifact. This one here looks pink, but we could use another tissue with another slide and another stain, and it may be purple, or it could be green, or it could be black, or any number of other things. So color is one of those things we want to be really, really careful about. But sometimes color can be useful, and that's what we see here. And I'm going to switch to my highlighter here. This is what we see here with our goblet cell. One of the key things to a goblet cell is a cell that produces a special protein, and that special protein is called mucin. Mucin is a protein that, when added to water, forms mucus. So your cells don't actually make mucus. What they actually do is they make a protein called mucin. They release that mucin into the lumen of the tissue, where it mixes with water and becomes mucus. All right, oops, I spelled that wrong. There you go. Forms mucus. All right. Now, mucin, most stains don't bind to mucin. So one of the ways we can often identify a goblet cell is the goblet cell is much lighter in color than the tissue that surrounds it. So if you notice, it's a little bit lighter in color here, and we'll see some even better examples of this when we look at some histology slides. So this cell is bulging out, and that's actually where it gets its name, goblet cell. As we'll see, and here's the illustration from your textbook, this cell produces a large amount of the secretion, filling it up so that the cell kind of bulges out like a goblet, like a big, huge goblet. And a big, huge goblet cell with all this bulging type. So it looks like a big goblet, drinking goblet. And so that's where it gets its name. So the very first potential type of structural classification is a unicellular gland. In fact, if we cheat, and I need a little space, so I need to juggle some of my stuff around, there are 8 specific structural classifications. And the first is a unicellular gland. So one possible type of structural classification would just be like this goblet cell, a single-celled gland. All right. However, the rest of our glands are going to be multicellular. With a multicellular gland, and there are 7 specific types. We'll do the rules first, then we'll go through all the pretty pictures and everything. There are 2 ways that we're going to describe them. All right. Now, remember, these are multicellular glands made up of 2 or more cells. So with multicellular glands, all multicellular glands have 2 parts. As we talked about, they have the secretory structures, which again make the secretion. And they have the ducts that carry the secretion outside of the body. So we identify them two ways, by their secretory structures and by their ducts. The first way we identify them is the shape of the secretory structure. Right? In the shape of the secretory structure, there are two main shapes. The first is a long, thin, narrow structure, tube-like structure, and in fact is called a tubular gland because it's a long, narrow, thin, tube-shaped structure. And the other is a short, round, bulbous, right, ball-shaped structure. And this round ball shape are what are known as acini, or also known as alveoli. So again, and those are the singular. Let's go back to plural since those are the plural. Let's go back. So acinus or alveolus, which is the singular for those. So we have tube shape and we have ball shape secretory structures, one of two shapes. And again, the term acini and alveoli can be used interchangeably. And fun with vocabulary. The second way we identify them is actually by the number of secretory structures and the number of ducts. And there are three main types. You can have one duct and one secretory structure. And if you do that, you are a simple gland. You can have one duct and two or more secretory structures. And you are a branched gland. Now, I will warn you that some textbooks use the term simple branched, but I find that confusing because the other one is simple. So if you just want to use the term branched gland, I am perfectly happy with that. And the third type is two or more ducts and two or more secretory structures. And if that is the case, you are a compound gland. All right? So let's let this information sink in for a second. And once this information sinks in for a second, we can actually take the time to draw these structures and make sure that it makes sense to us. All right?
Quick question.
Yep.
Do we have two ducts with one secretory?
Nope.
No, okay.
No, you would not have that. No. So these are your only three possibilities. Okay? So the good news is there really aren't too many curveballs on this one. So this one's going to be pretty straightforward. So let's go, now that we have our rules here, although I've got a sneaking suspicion that when I go to my whiteboard and come back, all of this is going to be gone. So hopefully you've got all of this, but we're going to go through it again. Let's go to our whiteboard, clear our whiteboard, and draw some glands. Now, we will start here with the surface of the body, because remember, all of our exocrine glands secrete outside of the body. All right? So, and for artistic purposes, we are going to, again, remember all of these multi-celled glands have two parts. And those two parts are the ducts and the secretory structures. And so what we are going to do to make this easy is the ducts I will color blue, and the secretory structures we will color red. So one possible structure that a gland could be comprised of is it could be a single duct, and connected to that single duct is a long tubular secretory structure. So here we have one duct, one secretory structure, and it is long, narrow, and thin. So how would you identify this structural classification of a gland? What would you say this gland was? There you go. Perfect. Excellent, Nicole. Simple tubular. Absolutely. This is a simple tubular gland. Let's try another one. We have a single duct, which connects to a single round ball shape secretory structure. What would this be? Come on, what's this one? Simple alveolar. Excellent. Excellent. Getting the sense of this, that works. Oh, I forgot one thing. Hold on, got to move this one out of my way because there's one more piece of information. All right, so far so good. We have two simple glands. One that is a simple tube, one that is a simple alveolar. One has a tube-shaped secretory structure, one is a ball-shaped secretory structure. We're comfortable with that? There is one catch with this tubed one. What can happen in it, and I'm just going to draw it as a line this time because, again, we understand how limited my artistic skills are. What can happen with the tubular one is that while sometimes the tube can be straight and linear like this, what often happens with the tube is that it actually coils and spins and wraps around. It doesn't go in a straight line. It kind of wraps around in a coiled sense. And in fact, that's what we call this. We call this a simple coiled tubular gland. So simple tubular glands can be straight or they can be coiled. So that we can put there and then cheat and move these over a little bit more. So this come over here. This can come over here, can come over here. Excellent. All right, perfect. Okay, so one little catch on the tubed one is they're often twisted around, so we just call them a simple coiled tubular, but still one duct, one secretory structure. All right, that's two. Let's do the third. So start first with a single duct, and off that single duct, I don't know if I'm going to be able to do this. I'm going to have to cheat and draw it. All right, excellent. Off this single duct, we can have, undo that, red. Two long, narrow, tube-like structures that connect to it. How would we identify this particular gland?
Simple branched.
Right, or again, remember, I don't like using the simple, but if you write simple branch, it's totally acceptable. But I find the simple is confusing, so branch tubular. But if you want to put simple branch, totally fine with that. That is absolutely 100% correct. But I'm just going to put branch tubular to help us to distinguish them. And then, of course, we could also have one duct, two or more, and in this case, let's go ahead and put three. Ball-shaped secretory structures that attach to that single duct. What do we have here?
Simple.
Yep, simple. Here, I'll
branched alveolar.
Excellent. Excellent. Perfect. There you go. All right. So far, so good? Everything making sense?
Yes.
Oh, wait, hold on. Let's go back to black for my surface of the body. Bingo. All right, next. You can have, oh, I don't have to draw this one, a gland that has two or more ducts connected to two long, narrow secretory structures. What would this be?
Compound gland.
Yep, but compound you'd only get partial credit for, because what's the other half of it? Compound.
Tubular.
Tubular
There you
gland.
go. Compound tubular. Excellent. This would be a compound tubular. Perfect. All right. Option, whatever the next one is, B. It's not really B, but you get the idea. We could have a gland that has two or more ducts. Let's give this one 3 ducts coming off of it. And each one of those connects to a circular so 2 or more ducts, 2 or more ball-shaped secretory structures. What do we have here?
Compound alveolar.
Excellent. So there's only one left, all right? Because remember, there's supposed to be 7 of these. These 2 count the same. 1, 2, 3, 4, 5, 6. There's only one more left. And again, there's always going to be that alphabet soup term you're going to have to worry about. And this is going to be it here. This very last gland has 2 or more two or more ducts. Let's give this one 4 ducts. But what's special about this one is this has both ball-shaped secretory structures, and it also has tube-shaped secretory structures. So what do you think we would call this one? Well, 2 or more ducts, 2 or more secretory structures. So definitely compound. That's the easy part because it's definitely 2 or more of each. But this one has both tubes and alveolar in it, right? So we need a way of saying both. And the way we say both is by mashing the 2 words together. This is a compound tubulo alveolar, all one word, tubulo-alveolar gland. So these 7 plus the 1 unicellular gland are your 8 structural classifications of exocrine glands. Now, again, I have done with my amazing artistic skills an amazing job of doing this, but for once, I would say that I'm a little better than the textbook. All right, doesn't happen often, but let's take a quick peek at the picture from your textbook. All right. Because notice your textbook does a nice job of talking about these. Here we see, like I did, where's my annotation? There it is. They have color-coded the duct and color-coded the secretory structure to differentiate them. And these are kind of long, narrow tubes. But those are the balls. Notice there isn't really that, it's not that much of a difference the way they draw between the alveoli and the tube. They don't really distinguish it quite as obviously as I do. But again, we have those simple 1-to-1 branched 1 duct to 2 or more secretory structures there. Here are the compound 2 or more ducts with 2 or more tube-shaped secretory structures, 2 or more ducts 2 or more ball-shaped secretory structures. And then here is our compound tubulo-alveolar, where we have balls and tubes together on one gland. All right. So again, my drawing may have been simpler, but I emphasized it a teeny bit better than the way you're looking at it. Now, notice when you have 2 or more secretory structures like this, typically this gland is going to make 2 or more substances. If you remember, we talked about saliva, and that's the classic example and the one we'll actually see histologically. Saliva is a very complicated secretion. It has serous fluid, has a watery component that mixes with your food and allows it to bind the food together as you're chewing it. But as we also talked about, you can knee on your younger sibling's chest and dribble a tiny bit of spit so that it almost touches their nose and then slurp it back up because it has mucus in it as well. So notice that has 2 components, a watery component and a mucus component. To make those 2 components, you need 2 different types of secretory structures. So in our salivary gland, what we're going to see is the ball-shaped structure makes the watery part, the serous fluid part of it, and the tubes actually make the mucin. And so the other way we're going to be able to tell them apart, not just by their shape, but by the composition of them, because they're going to be producing different things. So that's going to be an important thing we're going to note when we look at this histologically. All right. Perfect. All right. Any questions on the 8 structural classifications of our exocrine glands? Excellent. Like I said, I love it when everybody's quiet because that tells me that you've all mastered the material. And like I said, that means I get to make the tests harder. So perfect. I'm glad that all makes sense. Excellent. Let's talk then about the functional classifications. The good news, there are only 3 main types of functional classification. So there are 8 we need to learn for structural, but there's only 3 we need to learn for functional. And let's look at the pretty pictures for this to make some sense of this. Starting first with the Merocrine mode of secretion. Now, this is by far the most common type that is found in humans. And let's actually look at the pretty picture to actually see what is going on. Notice we start with DNA in the nucleus, where we're going to make our messenger RNA that goes to the rough endoplasmic reticulum, where we make the sequences of amino acids. They are then sent to the Golgi apparatus, which cuts them and bundles them and makes the functional proteins. And it then puts it into vesicles. And notice what happens to these vesicles. These vesicles move to the plasma membrane. When they get to the plasma membrane, they bind with the plasma membrane and they release their contents. What did we call this process of bundling these things up in a vesicle, bringing it to the plasma membrane and releasing it? What did we call that? Oh, wait. There it is, exocytosis. Excellent. That's what Merocrine glands do. Glands that use the Merocrine mode of secretion use the process of exocytosis. They produce the substance, bundle it in a vesicle, bring it to the plasma membrane, and release its contents. Now, is the cell damaged by the process? Is the cell damaged by the process of exocytosis?
No.
No, right? This is not damaged in this process. In fact, that kind of sounds like a silly question. We'll see why it's not a silly question in a minute, but it does kind of seem like a silly question. No, the cell is not damaged. The cell's not changed by this process, right? And so it's something that can be occurring continuously. And like I said, this is the most common type, right? Again, here we see a single cell, what it's doing. Here is the pretty picture from your textbook. Although I will point out one small error with this picture. Well, again, not an error, an artistic license that our artist has made. Notice here we see a ball-shaped secretory structure where we're producing the vesicles, and the vesicles are expelling their substance into the gland, and then it leaves out the duct. Although if you notice, our artist put a couple of vesicles within the cells of the duct. Are the cells of the duct producing and releasing secretions?
They shouldn't.
They should not, absolutely. So that is, again, that error that our artist has made in that. It is just the secretory structure that is producing them and releasing those contents. Remember, the duct is just a conduit for that. But other than that minor error, it's a pretty good illustration of how these Merocrine glands use exocytosis to release their contents. And like I said, it's the most common type. This is how your salivary glands, your sweat glands, your pancreatic glands, most of the glands of the body use this Merocrine mode of secretion, including the sweat glands, like these on the surface of our skin. These on the surface of the skin produce that watery sweat that evaporates to allow us to regulate our body temperature. All right? But there's a second mode of secretion. This mode of secretion is what is known as the epocrine mode of secretion. Again, let's look at the pretty picture and see what's happening here. Again, transcription, translation. We get that sequence of amino acids, which goes to the Golgi apparatus, cut, bundled, made into a functional protein, and put into a vesicle. But notice these vesicles don't release their contents via exocytosis. Instead, what happens here is the apical portion of the cell pinches off. When it pinches off, it is able to release its contents. It releases the secretion, but notice it also is going to end up releasing some of the cytoplasm. Because of this, this makes for a more viscous secretion, more viscous and organic secretion, right? Now, again, obviously in this process, the cell is damaged. Yes, right? It's harmed by this. Does the cell die? No, it doesn't die, but it is damaged by this. However, it is capable of regrowing, repairing, and as it grows and repairs, it makes more secretions, fills the apical part with the secretions again, and then pinches off a second time, and that process continues. So the cell is damaged, but it doesn't die. So it can repair and continue the process. Excellent. All right.
Question?
Yes.
When you said it's more viscose, what does that mean exactly?
Viscous means more thick, right?
Okay.
Not quite as watery.
Oh, got you.
Right. Let's go again to an example, right? Now, an important thing to remember here is epocrine is a functional classification of a gland, or the other way we can be thinking of these terms is mode of secretion, right? What I mean by that is how it releases its contents. That is what we're talking about. Does that make sense to everybody? Dead silence, that means yes. Excellent. I make a point of emphasizing, excellent, thank you, Michelle, that's exactly where I'm going right now, is because let's go back to our sweat glands. As I mentioned, on the surface of your arms, if you got up and ran around your room 16 times, your arms would get all watery and sweaty. You, as an adult, would also probably get a little stinky. However, you know, your 6-year-old kid can run around the room for hours and still smell like a rose. Why? Well, it turns out we have a second type of sweat glands. These second type of sweat glands are associated with our secondary hairs. So areas like our axillary region or our groin region or the thoracic region, right? Buccal region. these glands produce a more organic type of sweat. These are the ones that the bacteria love and can produce body odor if it accumulates and things along those lines. And because this is a sweat that isn't so much involved in body temperature regulation, but is more actually involved in reproduction. They've actually done studies where the scent, again, we do not have pheromones as human beings, but the scent of the substance that is produced from these glands does have a typically perceived as pleasing type of sensation associated with them. Now again, once the bacteria gets in there and breaks them down and you have the bad body odor, then nobody enjoys that. But the scent of them is somewhat pleasing organically. And so because of this, they assumed these cells use the epocrine mode of secretion. And so in our skin, we have glands called epocrine glands. Oops, why is that not writing? In our skin, we have glands called epocrine by name. Of course, they were called epocrine by name because they use the epocrine mode of secretion. And being the smart, sophisticated people we are, we use this to our advantage. Back in ancient times, there was a basketball player by the name of Michael Jordan. Luckily, because ESPN just did a huge 10-part series on him, everybody now remembers who Michael Jordan was. But in ancient times, he was an amazing basketball, far better than anything LeBron James could ever hope to be. That being said, one of the things that made him so amazing wasn't just how great he was as a basketball player, but he became a true force in advertising. And everyone he wanted to be like Mike. In fact, one of the things they actually did is they actually collected some of the sweat from the epocrine glands of Michael Jordan to make an aftershave. So that you, as a poor mere mortal, might never be able to play basketball like Mike, but you could actually smell like Mike by taking those epocrine gland secretions and using it to make an aftershave. Amazing stuff. Tells you about the popularity of sports, science being used for advantages, and all these kind of things. There's only one problem with all of this. And the problem with all of this is that many years later, as we continued to study these glands, they learned that these secondary sweat glands that are used by your body actually use the Merocrine mode of secretion. What this means is, I say next week, but really I mean Thursday and Friday. When we talk about the skin, we are going to talk about glands that are called epocrine glands because they thought they used the epocrine mode of secretion. But guess what? They don't use the epocrine mode of secretion. Now, as soon as they found that out, did they go and change the name of these glands? Nope, they did not. So we are going to learn about epocrine glands in the skin that are called epocrine by name, but actually use the Merocrine mode of secretion. Welcome to Anatomy and Physiology. All right. In fact, there are no confirmed examples of epocrine glands in humans. If you want, again, so a couple people asked for an example. If you want a true example of an epocrine gland, the best example of that are actually the anal glands of dogs. Right? When a dog meets another dog, what's one of the first things it does?
Sniff each other?
Yeah, they sniff each other's butts. The reason they're doing that is because dogs have an anal gland that uses the epocrine mode of secretion that tells them pretty much everything about that dog. What their overall health is, what they've been eating, where they've been. They get a tremendous amount of information from this organic secretion that is produced by those anal glands. Now, notice I said no confirmed examples of epocrine. Oops. Epocrine in humans. There is research currently going into the mammary glands. If you think, and we'll talk about the mammary glands in more depth later, they produce breast milk. But there's really two types of breast milk. There is the foreign milk, which is much more watery and has much more carbohydrates in it. and there's the hind milk, which is a little bit thicker and has more fats and proteins in it. So again, there's two different compositions to breast milk, and it is believed that some of those glands may use an epocrine mode of secretion, but it has not been confirmed yet. But there are some people who think that way, but until it is confirmed, we're not going to make that mistake again. So as of right now, we'll say there's no confirmed examples. But if we cheat, we clear all the pretty pictures and go back to the original slide. Notice for epocrine, they do actually show the mammary gland as an example. We just don't know that for certain. It is believed that it may be the case, and they do a nice job of showing how the top pinches off and regrows and starts again. But that has not been confirmed. So as of right now, there are no confirmed human examples. We believe it may be the mammary gland. We know anal glands of dogs use the epocrine mode of secretion, but there's no confirmed human examples. We think there may be some in the mammary glands, but we're not certain. All right. And that brings us to our third functional classification, or again, we could also use the phrase mode of secretion. And this third mode of secretion is called the holocrine mode of secretion. Now, let's look at the pretty picture here again. Notice in our pretty picture, it's showing us a stage of cell division. What stage of cell division is being represented here? Oh, wait, anaphase, perfect, it is anaphase. However, notice they got the V's pointed in the wrong direction. Again, fun with those. You gotta remember, the artists who do this are often not necessarily anatomists. So they notice they've got the V's pointing in the wrong direction of our anaphase, but you've got the right idea. What we have here are cells that divide mitotically. As they divide mitotically, as you can see, they fill up with their secretion. So that's what we see going on over here. We see that it's accumulating the secretions. And then when the cell reaches the top, it bursts. It ruptures. It bursts, releasing all of its contents. Not just the secretion, but the entire cytoplasm is released in this process. Does that damage the cell? Yeah, absolutely. That kills the cell, right? This definitely damages the cell. It kills the cell, but luckily we are making more behind it. Now, again, notice we've got this picture here. The pretty picture from your textbook is better. So let's look at that. Again, notice here they've got the V's pointing in the correct direction for our anaphase. We are dividing cells, producing new cells. They get to the top and they produce their secretion and fill up with that secretion and then rupture, releasing all of its contents. This produces a thick, very organic secretion. Right? And the classic example of this are what we commonly refer to as our oil glands of the skin. But as we'll learn, the correct name for those are sebaceous glands that produce not oil, but actually a substance called sebum. And as you know, because now everybody's washing their hands for 20 seconds, 17 times a day, when you're constantly washing your hands and removing that sebum, what ends up happening to your skin?
Dryness.
Yeah, it gets dry and it cracks. So that oil helps to maintain the integrity of the skin, helps to maintain the integrity of the hair, right? Help to perform that protective barrier for us, right? This secretion is controlled, again, heavily by hormones. So when we reach puberty, one of the things that happens is that those puberty hormones, those growth hormones like androgens and estrogens, cause an increase in the production of this stuff. And as there's an increase in the production of this stuff, one of the things that can happen is that these ducts can get congested with that thick, viscous, organic material, and it becomes clogged. And it's clogged with this warm, moist, organic material that bacteria just love. So a bunch of bacteria come in and start feeding on this, and this area becomes infected and inflamed. And what do we call that condition?
Acne?
Acne, exactly. These are the cells that are, and that thick, viscous, organic secretion of the sebum is what causes that acne. All right. So there, those sebaceous glands are the classic example of this holocrine mode of secretion. All right, so there you go. Those are our three functional classifications or modes of secretion. And we also identified the 8 structural classifications for our exocrine glands. Questions on that? All righty, with that, how are we on time? We're doing good, excellent. With that, we are done with all of the lecture portion that I wanted to do for the epithelial tissues. What we need to do now is switch gears and do a little bit of the histology, so we can start looking at our handout and you can start figuring out what it is that we need to know and how you're going to attack this material. So we're not going to have a time to go through all of them together, but there are some key things that I want to point out on these things as we go through them. Both what you're looking for, what you should be labeling, how you should be labeling it, and most importantly, how I'll be asking these questions on the exam. And then the other thing, and then we should still then have enough time to start talking about tissues, pardon me, about connective tissues, so we can start talking about our connective tissues so that we can spend as much of the next 2 days as possible talking about the skin. All righty, questions on any of that? All right, let's come back and do histology, but the histology is, like I said, where things start getting dense. So let's go ahead and take our next break. It is 10:31 right now, so let us return at 10:46. And we will restart the recording at that part. So any questions before we take our break? All right, I will meet you back here then in 15 minutes. Let's go ahead and get started. Before we dive into histology, and hopefully, like me, you have your cheat sheet in front of you, because we're going to keep it really simple this first time through. Just go in order and make sure that we understand everything that we are looking at. I know the temptation when I throw these slides up is for you to furiously be coloring all these things in or drawing these pictures that you're seeing. But if you're busy furiously trying to copy what you see on the screen, you're not really listening to what we're talking about. So like I said, for this first time when we go through, I really encourage you to just sit back, watch, listen, think, look at your list, interact with me and answer the questions that I ask. Because again, I'm doing this for your benefit. If you guys aren't interacting with me, I know this stuff already. I don't need to go through this. We're doing this for you. So do that. And then either when you watch this the next time, or better yet, when you're using your histology atlas or your textbook, your lab manual, or the practice anatomy lab, or whatever resource you're using to study these tissues, you can then do the drawings then. All of these pictures are things that I got off of the almighty interwebs. You know, good almighty Google is the one that gave me these. So again, I found all of them that way. You can find plenty of examples of them this way as well. But remember, as I mentioned, I encourage you not to start with the Google search, because like I said, when you type in ciliated, pseudostratified, columnar epithelial tissue, you're going to get a lot more than that on your search when you do that for the images. And if you don't know what you're looking at, then you're not going to be able to comprehend it. If you're more familiar with it first, then you'll be able to more readily tell what is and is not accurate. And that way you save time studying this material. The other thing I forgot to mention at the beginning of class today, but now is a good time to remind us because we're going into the lab stuff, is I don't know how many of you took advantage of the open lab last week, but I want to remind you again tomorrow is your open lab with Jeff to be able to ask questions and get information from him. You can ask him about my testing policy or what he thinks of that. I'm sure he'll give you answers to that. Remember, you get the extra credit for going to those open labs, but more importantly, the knowledge you gain is going to help you to be much more successful. And I will remind you that tomorrow is the last open lab before your first exam. So if you have not gone to an open lab yet, I encourage you to check it out and see how that goes so that you can get that information from Jeff to help you to be successful on the exam. All righty, with that now, and I want to switch back to my pointer, let's take a look at what we would be looking at our first slide. Now, unfortunately, again, in the classroom, I have the slides. I can change the magnifications. I can change the views. This was something that I threw together so that we could at least talk about these things. And so we're going to start actually with a high magnification view. And like I said, these were thrown together quickly. They aren't necessarily indicative of what would be on the exam. This is slightly out of focus. So obviously for the exam, I'd like something hopefully a little bit more clear. But in my haste, this was something that I found that I thought was pretty good. And as we look at this tissue in its entirety, my first question to you is to identify the specimen in the field of view. And what is your answer to that question? Again, remember, I'm doing this for your guys' benefit. If you don't interact with me, if you don't answer the questions, then we stop and we just move back to lecture. So what is the specimen you see here in the field of view?
I have no idea.
Anybody have an idea?
Is it a cell going through mitosis?
No, not a bad guess.
Possibly.
What might the specimen be in the field of view? If only I had a handout that had a list of all the things I was going to be responsible for. And if maybe the very first thing on the list happened to have the word specimen in parentheses next to it, then I might be able to figure out what the specimen in the field of view is. What's the specimen in the field of view? There we go. I got a couple answers. Kidney. Excellent. It is the kidney. And of course, how did you know it was the kidney? Ryan, how'd you know it was the kidney? Right. Well, yeah, absolutely. Because it had specimen in parentheses next to it. And it was also the very first thing on the list. Kind of convenient that the very first thing I asked was the very first thing on the list. All right. Now, obviously, these are not going to be presented in order. So when it actually comes to the test, there's got to be a way that you're able to readily identify this as the kidney. Right? That's going to be a key to a lot of the histology. A lot of histology, because these are timed exams, are finding quick and easy ways to readily identify something. When it comes to the kidney, the quick and easy way to identify it is because this structure here, and red's not going to work for that, so let's undo that. Now let's see what green looks like. That's a little better. This structure inside of the circle. No, great question. I specifically did not pick pictures out of your histology atlas or your textbook or your lab manual for the studying purposes. Because again, I don't want you just memorizing those pictures. Those are great starting points for these things. That's a great starting point for these things, but I want you to be able to recognize them on any obvious example of these things. So that's why I purposely didn't use any of the pictures from your textbook, any of the pictures from your atlases, any from the pictures of your lab manual, or anything else. Like I said, I found all of these on the almighty Google. All right, and excellent. Yes, to answer, the epithelial tissue handout is on is in your modules under the lab handouts. So you have that there, okay? Now, the reason you can tell by looking at that this is a kidney is because this structure inside of a circle is a unique structure that is found in one place and one place only in the body. This unique structure is only found in the kidney. And what do you think this structure is?
The renal corpuscle.
The renal corpuscle, absolutely. And I'm going to go ahead and erase the arrow, and I'm going to switch to my highlighter instead. This renal corpuscle is made up of 3 components. The first component is this thing right here, this big squiggly knot thing looking here, this big, weird, squiggly thing is a very, very special capillary. This very special capillary happens to, not surprisingly, have a name, and the name of this very fancy capillary is the glomerulus. Oops, cap lock off. Now, do you need to know that?
No.
No, because it's not on your list. So you don't need to know that word. So feel free to ignore it. But what we do need to know is that this is a big fancy capillary. The second component of it, and I will use the highlighter again, and this time we'll use brown, is this thin layer of cells that forms the outer boundary of this structure. So here we have a layer, and any idea what we might call that layer?
Bowman's capsule.
Bowman's capsule, excellent. Now, as it turns out, Bowman's capsule, as you can see, is made up of a single layer of flat cells. So based on that, what tissue type is this structure made up of?
Simple squamous epithelial tissue.
It is a simple squamous epithelial tissue, excellent. And this dark spot right here, what organelle is that?
Nucleus.
Nucleus. There you go. So notice our renal corpuscle is made up of three things. Thing one is the layer, Bowman's capsule, made up of a simple squamous epithelial tissue, right? This is a desk, it is made of wood. This is Bowman's capsule, it is made of a simple squamous epithelial tissue. Part two is this special capillary, a blood vessel that is located in here. And part three is the space between them. And what do you think we call the space in between the glomerulus and Bowman's capsule?
Bowman space.
Bowman's space, excellent. Now, why are these things called Bowman's space and Bowman's capsule? Just out of curiosity.
Is it actually science?
Yeah, good old Bob Bowman was the very first person who identified these things, drew them, submitted them to a magazine, right? Planted his flag in it, and because he planted his flag on it, he got to put his name on it, right? There's a lot of antiquated terms this way that are based on the anatomist to identify them, and those names are pretty meaningless, but other than giving fame to the person who discovered them. So you will notice in the textbook, Bowman's capsule is also referred to as the glomerular capsule because it goes around the glomerulus, or the renal capsule because it is part of the renal corpuscle, and any of those are acceptable names. All right? So there you go. This is our structure. Now I'm going to cheat and erase my highlighters so we can see that again. And again, I want to emphasize here, I guess we'll do it with a highlighter. That again, this is Bowman's capsule and it is a simple squamous epithelial tissue. Now, as a simple squamous epithelial tissue, like all epithelial tissues, does it have an apical surface and a basal surface?
Yes.
But with a simple straight line, is that something that's pretty easy to see and identify? Not really.
No.
Right? So that's pretty challenging, but technically it's there. Also, as we know, the other, if we think about it, Bowman's space is a lumen, if you think about it, because it's enclosed. And also, because it is above the apical surface, it would also be a free space. Now, what I would say is on the exam, if I have this arrow pointing to this space like this, and I ask you to identify this space, while lumen and free space are accurate answers, is it as specific as we could be?
No.
No. So you would get partial credit for lumen or free space, But because this one has a specific name, you would need to say Bowman space for full credit. Okay? Now, of course, the last thing we need, and again, I wouldn't say this is necessarily something you need to know for the exam because I'm not necessarily going to ask this on the exam, but we do need to kind of know what the function of this is. This is inside the kidney, and its job is to filter the blood. What happens is stuff gets filtered out of the blood, out of the glomerulus, and Bowman's capsule is basically the cup that catches it. It catches it in Bowman's space, and then it goes to all of these tubules, like this one right here, all of these tubules in the kidney to be processed. You have 1/2 mile of tubules inside your kidney processing this filtrate. You produce 200 liters of filtrate every day. You certainly don't produce 200 liters of urine, and that's because these tubules in our kidneys are going to filter and process it and reabsorb most of those materials. So, as we see the renal corpuscle, we now see this thing that I've put my circle around. And what do you think that structure that I've put my circle around might be?
A renal tubule.
Perfect. It is indeed a renal tubule. Excellent. It is a renal tubule. Notice this one is cut in a cross section. This one over here you can see is kind of cut in a longitudinal section. This one's more oblique, some more cross sections. And this renal tubule, being a tubule as we talked about, what type of cells is it comprised of? What tissue type is this?
Simple cubital.
Simple cuboidal. Excellent. And notice, conveniently enough, with a thicker tissue, now it's more easy for me to, actually let's clear the circles first. I could draw an arrow here, and I could ask you 2 questions. Question 1, identify the surface. And what would your answer to that be?
Apical cell?
Well, so remember, we take a epithelial tissue with its apical surface, and when we roll it up into a tube and form a lumen, the apical surface is on the inside. So this is actually the
Basal
basal surface. This is the basal surface. I could also ask you the question, identify the extracellular material found in this location.
Basement
membrane.
Basement membrane. I could move my arrow here and ask you to identify this surface. What surface is this?
Apical.
Apical. Identify the space.
Lumen.
Lumen or free space. Remember, it's always going to be free space. Free space is always going to be the right answer for this. In this case, the free space is rolled up, so it becomes a lumen. But lumen is absolutely the correct answer. Absolutely perfect. And I think it's a little better here. Identify the organelle.
Nucleus.
Nucleus. There you go. See how simple this is?
Now, different colored pointer. It was hard to see that one because I thought you were pointing somewhere else.
Sorry. Yep, I can do that. Let's clear that one. Well, here's the other thing we're gonna do. We're gonna switch slides. But let's see if I can get a better arrow. Let's do that. Let's do that. And I just worry the black's not gonna contrast right. What does that look like? Is that a little better, easier to see? Perfect. Excellent. Identify this structure.
Stomach?
It is indeed another renal tubule, right? Normally, I would have shifted the slide and you would know we were still looking at the kidney. But you are correct. This does, in fact, happen to be the kidney. However, notice this picture here, we don't see a renal corpuscle. So there isn't anything that guarantees us that this is indeed the kidney. So in this case, if you had just said tubule, that would be a perfectly acceptable answer. And here's the other thing too. Remember before when I was back at the previous slide, let's go back to the previous slide. Move my arrow out of the way. Whoops, nope, wrong button. Remember, I could ask you identify the specimen. And because you saw the renal corpuscle, you knew this was the kidney, right? Because that's the dead giveaway, you're looking at the kidney. Do we have a dead giveaway here that tells us this is the kidney? I'm the dead giveaway. I've told you it's the kidney. But as you look at this picture, is there any place here that is the dead giveaway? Do we see a renal corpuscle anywhere on this?
No.
No. So what I would do instead, the other way I could ask that specimen question is to ask it this way. I could point at this tissue and I could ask you, identify one location in the body where you would find this tissue. And what's one location where you would find a simple cuboidal epithelial tissue?
Kidney.
Kidney. There you go. So notice I could ask that question both ways. I could ask, identify the specimen, where you're able to tell me what it is by some dead giveaway in the picture. Or I could ask you to identify a place in the body, a location in the body where you would find this specific tissue, and then you'd be able to answer it that way. All right. Now again, one of the reasons people don't one of the reasons people lose points on the exam is not because they don't know the information, but because they don't read the question carefully. Read the question carefully so you can answer it correctly. Notice, as it is right now, there is, I think off the top of my head, I can think of 5 questions I could ask with this arrow on this slide right now. So let's go through them. Identify the structure.
Renal tubule.
Renal tubule, or just tubule, remember. For this one, tubule would be finest top. Identify the tissue type.
Simple cuboidal.
Cuboidal epithelial tissue. Excellent. Remember, you want to say epithelial tissue too. Simple cuboidal epithelial tissue. Excellent. Identify the surface.
Apical.
Is that the apical?
Basal.
Basal, right? Identify the extracellular material you would find in this location.
Basement membrane.
Basement membrane. Was there anything else we could say instead of basement membrane? Basal lamina. Excellent. Remember, on the exam, you may put either of those 2 terms, right? Again, if whichever one you want to spell, whichever one you want to use, you're welcome, because both are 100% acceptable. However, both are vocabulary terms that you need to know. So if I use basal lamina, if you decide I'm going to write basement membrane has made sense to me. If I use the term basal lamina in a essay or multiple choice question, you definitely need to recognize that and know what it is, okay? So as long as you recognize the terms, you may use either one to describe these things on the exam.
But they mean the same thing, correct?
Exactly, just like anterior and ventral mean the exact same thing. Like I said, that's part of the problem with anatomy and physiology. So many people study this in so many different ways, there's a different vocabulary and often more than one term that means the exact same thing.
Now, quick question.
Yes.
Is there an example where you would use basement membrane and basal lamina as a specific to that place where you cannot mix them, like choose one, you know?
Okay, so here's what I will say. If you've bothered to read the text, so here's a good general rule for this class. This is a good general rule to remember for this class in general. Right, so in this class, sky is blue, right? What do I mean by that? Well, my daughter, who gets woken up every morning by me talking to you guys because we share a room. I mean, we share a wall. We don't share a room. That'd be weird. We share a wall, and I talk loud, is 12. And if I were to go over there and ask her what color the sky is, what would her answer be?
Blue.
She would say the sky is blue. But at night, is the sky blue? When it's raining, is the sky blue? At sunset and sunrise, is the sky blue?
No.
Does that mean my daughter's stupid? Well,
No.
She's a little off. But what it means is when she says the sky is blue, is it is a simple, basic answer that is right most of the time, but the truth is it's far more complex than that. one of the things you have to remember is everything in this class is far more complex than we have the opportunity to go into. Every single topic in this class is something we could probably literally spend an entire semester talking about, all right? So what we're having to do is simplify things a little bit. If you've looked in your textbook, what you will see is that the basement membrane of a cell of an epithelial tissue is actually made up of a couple different layers of material. We're not going to worry about differentiating that. So for our purposes in this class, basal lamina and basement membrane are going to mean the exact same thing. All right, so we are, yeah, it's true, probably true. It is true for every science class. Every science class, you are learning the sky is blue. When you get to your pre-med, when you get to your med school, when you get to your kinesiology, when you get to nursing program, that's when you get to learn about when the sky is gray and the sky is purple and the sky is black, and you get to learn about all of that. But here at A&P, the sky is blue. All right, so for us, basal lamina, basement membrane, completely interchangeable terms. Okay, great question. All right, and notice we went through 4 questions. I never got to the 5th. Identify a location in the body where you would find this tissue.
Kidney.
So there you go. So notice, and again, as you look at the list, you see that for every one of those questions, I use the specific word that was in parentheses. I will be giving you the cue to tell you which precise piece of information I'm looking for. And again, I'm not trying to be tricky. We have to be precise. And so that's why, like I said, I try to be very standard. That's why I've got this set up the way that it is, so you know exactly what to expect from me. And then, of course, lastly, let's move our pointer, identify the surface.
Basal.
Basal? Remember, there's nothing above this surface, so this would be aqueous. And identify the space.
Basement membrane.
Lumen.
Lumen, or again, free space. That would be acceptable as well. Perfect. Oh, and again, because it's on the list, identify the organelle.
Nucleus.
Nucleus. Perfect. All right, questions on this tissue? Excellent. All righty. Identify the tissue type.
Simple columnar.
Columnar. Excellent.
Epithelial tissue.
Yep, excellent. Perfect. Even better. Simple columnar epithelial tissue. Excellent. Identify the surface. Now this one's a little tricky. What you got to see is that this tissue has been kind of folded in on itself. So what we actually have here, if you notice, here is the space. There's the lumen. So notice we have the apical tissue of this one against the apical tissue of this one. Basically what's happened is the 2 apical surfaces have been folded close to each other, right? And so that's what happens. So again, the lumen isn't big, but this is still the free space. And here's the other way we can tell. Let's go back, put the arrow back, and let's ask a different question instead. Identify the organelle. What organelle is this at the tip of the pointer? It's not on our list, but we did talk about it. Oh, there it is, microvilli. Perfect. Remember, not cilia, microvilli. This is simple columnar. Notice all the cells are very uniform in their shape. All the nuclei are lined up like ducks in a row. So these are those non-motile microvilli. And remember, the microvilli are going to be on the apical surface. Here, identify this surface. Basal surface. And the extracellular material you'd find in this location? Excellent. Perfect. Organelle? Nucleus. Excellent. Identify one location where you would find this tissue.
Lungs?
No, not lungs. Where did we say we'd find the simple columnar? Stomach, excellent. That's one location. But what's another one? No, not kidney. Careful with esophagus. If you're going to say esophagus, you have to say the inferior third or the distal third, the bottom part of the esophagus. Because remember, the top is going to be the non-keratinized stratified squamous. Excellent. Small intestine and large intestine. Perfect. Remember, this is the one that is in most of the digestive system. Not all. The two ends of the digestive system are special, but everything else in between is this simple columnar. Now, I know the slide says stomach as the specimen, but as it turns out, this one is actually from the small intestine. So this one happens to be from the small intestine, but it's all the same thing. Now, answer two other questions for me. Identify this cell. Yeah, this is a goblet cell. Excellent. Identify the substance at the tip of the pointer.
Mucin.
Perfect. Mucin, right? And again, I don't care how you pronounce it as long as you spell it correctly. M-U-C-I-N. And remember, it actually produces that protein mucin. And then that mucin, then when it mixes with water, becomes mucus. And that's actually what this is over here. This over here is actually a little mucus inside of the lumen of this organ. All right. Perfect. I think that is everything on the list for this one. But
question.
Yes.
The goblet cell, where is the nucleus? Does it have one or?
It does. Again, what you got to remember is that this is a two-dimensional slice through a three-dimensional structure. Think of it this way. If you threw an apple in the air and just with your eyes closed sliced it with a knife, are you always going to get it perfectly in half where you see the entire core perfectly in the center of it?
No.
No, and that's what happens here. Notice this particular goblet cell, we actually see the nucleus at the bottom of it. Remember, this is it at the bottom of it as it enlarges. Here we see a little bit of this one's nucleus, but notice this one's nucleus is much more clear. Because remember, what this cell does is it produces its secretion and it fills this huge space, forming this big bulbous goblet cell shaped cell, right? That's where it gets its name. So yes, this one, you're absolutely right. The nucleus is at the bottom. This one here, though, is more clear. This one, we're only really seeing a part of its nucleus because it was only the way it was sliced. Notice different magnification, but this is the identify. Well, let's just go through it. Identify the tissue type.
Simple columnar.
Yeah, again, simple columnar. Again, notice tall, fairly uniform cells, nuclei all kind of line up like ducks in a row. Again, notice we have another nice goblet cell that we can see here. Clearly see the basal surface, clearly see the apical surface. And what organelle was this again?
Microvilli.
Microvilli, excellent. Right, and you can see it wrapping around the surface of one of those finger-like extensions of the small intestine. Here is our simple columnar. Compare this to this. What's this one here? Identify the tissue type.
Pseudo.
Ciliated pseudostratified columnar.
Excellent. See, look at that. It's already starting to roll off the tongue. By the end of the semester, you'll be saying it like nothing, but that's awesome. Absolutely. Ciliated pseudostratified columnar. Make sure you have all three parts. If you want to go pseudostratified ciliated columnar, you want to put columnar first, whatever it is, yes. Ciliated pseudostratified columnar, ciliated pseudostratified columnar, ciliated pseudostratified columnar. I can do it. By the end of the class, you will be able to say it 3 times fast as well. Excellent.
Question.
Yes.
What's the ciliated part?
Ciliated are the cilia, the organelles that are on the apical surface. Right?
Now, if you didn't have the cilia, you don't say the cilia part anymore, correct?
Correct. But the good news is there's no such thing as a non-ciliated pseudostratified columnar epithelial tissue. So if you have a pseudostratified columnar, it is always going to have cilia.
Gotcha.
Cilia is how you're going to recognize it. All right. So
And the
again,
cilia, is it the microvilla or the?
no. Remember, cilia and microvilli are both organelles that extend off the apical surface of the cell, but they're different. Cilia like this are typically longer, and remember, they're motile, they move. Microvilli are typically shorter, and they're non-motile. They're just increasing the surface area. Remember, cilia and microvilli are not interchangeable terms. They're two different organelles, non-membranous organelles, that extend off the apical surface of cells.
Got it.
Excellent. So I think we already answered the question, but again, beating dead horses is always a good thing. Identify the surface of the cell, where the arrow is pointing now. Apical, excellent, apical. Identify the organelle.
Cilia.
Cilia, excellent. Identify the surface.
Basal.
Identify the extracellular material you would find in this location.
Basement membrane.
Excellent. Identify the organelle.
Nucleus.
Nucleus, excellent. Identify one location where you would find this tissue.
Trachea.
Trachea, excellent. And where's the other one? Nose, there you go, nasal cavity. Perfect. Excellent. All right. There you go. I think that is everything on this one. This can have a goblet cell. This particular slide, I don't think I have another one. Yeah, this particular slide doesn't have a goblet cell in it, but it can have a goblet cell in it. The one from lecture, if you remember, had the goblet cell in it as well. So this particular slide doesn't have a goblet cell, but ciliated pseudostratified columnars can have goblet cells. This particular slide doesn't show it, probably off to the side somewhere. All right, excellent. Just that simply, we have been able to identify all four of our simple epithelial tissues. And now we can talk about our stratified. Now remember, there's only three we're responsible for. Well, really two, but one of those has two flavors to it. Remember, histologically, you're not responsible for the stratified cuboidal or the stratified columnar. All right? Now, I also want you to do one more thing. Notice here with our ciliated pseudostratified, as we look down here inside the box, we see a bunch of nuclei stacked on top of each other. All right? Notice when we switch to the next slide, our magnification changed a little bit, but we can still look at it. Notice when we look at the bottom, if we just look down here at the bottom, it basically looks exactly the same. It looks like a lot of nuclei stacked on top of each other. So notice if we look at the basal surface, we're not going to be able to tell these two tissue types apart. But when we go here, we have this dead giveaway at the top, and that dead giveaway at the top are the cilia. Well, it turns out when we look at the apical surface of this, we have a dead giveaway as well. Notice we have a bunch of flat cells up here at the top. And if we have a bunch of flat cells up here at the top, guess what type of tissue this tissue is?
Is it the skin? Or?
No, I didn't say the location. I asked you for the tissue type.
Keratinized stratified?
Well, let's start with stratified because it's definitely more than one cell layer.
Non-keratinized?
Well, let's not worry about keratinized and non-keratinized yet. We'll get to that in a second. All right. Stratified squamous. Excellent. Epithelium. And again, remember, you can use the noun epithelium. You could use the adjective epithelial tissue. Either of those are fine. All right. So we can clearly see this one definitely does have multiple layers of cells. And the cells on top are flat. But as you guys very astutely pointed out, we know there are two types of stratified squamous. There is keratinized and non-keratinized. And as someone also mentioned, there's the skin. And as we talked about, what do you know about the top layers of your skin again? What did we say about those cells in the very, very top?
They're dead.
They're dead.
They're,
The reason they're dead is they're filled with that special protein called keratin. What these cells do is they make so much keratin, they fill themselves up with keratin that they're all, you know what? I don't need this nucleus and all these organelles anymore, and they chuck them, and the cell dies. So these cells on the top are dead and don't have a nucleus. Well, what do you notice about these cells at the top? Do these cells have a nucleus? Yeah.
No.
No, that's what these big, long, flat, black things are. These are the nuclei. So you can have two things. You can have a nucleus or you can have keratin. You can't have both. So if these have a nucleus, then that means they don't have keratin. And so as you guys correctly assumed, this is a non-keratinized stratified squamous epithelial tissue. So this one is the non-keratinized stratified squamous epithelial tissue. And notice, we couldn't tell that by looking down here at the bottom. We could tell by looking up here at the top. All right. Actually, Ryan, that's my question for you. Identify one location where you would find this tissue.
Could be.
Skin.
Ah, there you go. Mouth, cheek, inside the cheek, right? The surface of your tongue, your pharynx, your throat, the proximal part of the esophagus. And remember, all of the moist openings to the outside world. That means the vaginal canal, that means the anus, that means the urethra. All of those openings to the outside world need protection, need to maintain moisture, and so they use this. Well, the nose is tricky. Right where you first put your finger in when you're picking your nose, the very, very opening has this in it. But remember, the rest of the nose is making that mucus and moving it. So the nasal cavity, remember, is our ciliated pseudostratified columnar. But the very, very opening does have a non-keratinized stratified squamous. All right. Now, of course, we can still ask all the questions we were asking before. Identify the surface. Come on, these should be getting easier. Identify the surface, apical, excellent. Identify the space.
Lumen.
True, free, although in this picture it's a little tricky. We don't really see the enclosed lumen, but we know if this is inside the mouth or if this is inside the anus or inside the vaginal canal, it's gonna be enclosed. But so lumen would be a correct answer, but free space is also a safe answer as well.
Question.
Yes.
How did we, since we can't see the lumen, how did we decipher its apical surface?
Because remember, regardless of whether you can see the lumen or not, the apical surface is always the top, and there's always nothing on top of an epithelial tissue. So there's always a free space above an epithelial tissue, right? So notice the surface of my skin has an apical surface that is just filled with air above it. If you take an epithelial tissue and roll it up into a tube, then you get a lumen. But it's still, that lumen is a free space. It's a hollow space above the tissue. So it's always going to be a free space.
We were pointing, the pointer was where the nucleuses really have,
Well, I pointed down here. I only raised it a little bit. So here it's a little bit, and again, because the words. So here, if I did it there, again, and on the exam, I try to be obvious. This is more obvious. Identify this space.
Gotcha.
All right. But you're right. I could point it here and ask for the organelle.
Nucleus.
Excellent. Surface.
basal.
So
Surface. Extracellular material you find in this location.
Basement membrane.
Basement membrane. Excellent. And notice, you can clearly see the difference between the connective tissue underneath and the epithelial tissues. Remember, we said epithelial tissues are very cell dense, whereas we'll see connective tissues are not very cell dense at all. All right. Now, I'm going to leave my box here for a second, although we'll see how it lines up when I switch slides. I need to erase. I need to erase that. Excellent. Now, again, very different magnification, but if we again just look down here at the bottom of this, we see a large number of cells packed all tightly, closely together. This doesn't look any different from what we just saw before, doesn't look any different from the ciliated pseudostratified. So where we need to look is towards the top. And there are two things that we need to, whoa, that's weird, cool. Two things we need to notice when we look at the top of this tissue. Now, the first thing we need to do is as we look at these cells, in any of these cells up here at the top, do you see a nucleus?
No.
No. Notice these cells, all of these cells have plenty of nuclei, but none of these cells at the top have nuclei in them. Why do you think they don't have nuclei in them?
Because they're dead.
Because they're dead and they're filled with what instead?
Keratin.
Exactly. So this is our keratinized. Stratified squamous epithelial tissue. And I will tell you my secret to remembering this. If I'm trying to remember keratinized stratified squamous epithelial tissue and what it is, I remember what it is. It is a keratinized stratified squamous epithelial tissue. When? Because again, you've got tomorrow off. So since you have tomorrow off, and because the flights are super cheap now, if you wanted, you could fly to England. And maybe if you got lucky, you'd get to meet the Queen. And when when the Queen came up to meet you, she would put her hand out. And what are you supposed to do when she puts her hands out?
Kiss it.
You kiss it. There you go. K-S-S-E-T. Kiss it. Right? The skin, you kiss. Keratinized stratified squamous epithelial tissue. Right? What about the inside of her mouth? Should you kiss that?
No.
No, that's the non-keratinized. The skin, the outer part, that it's okay to kiss. The Queen won't mind that. She'll get upset if you kiss the inside of her mouth. Right?
Now, question.
Yes.
For the squamous part, right? Do you know it because it's, you know, you're looking at the picture and you see that it's kind of like squished together, you know, like smashed down?
Squamous means flat. Exactly. These are flat cells.
Okay, okay.
Now, I do have one other question. Notice these cells are not attached to the top of the tissue. They're kind of floating in space. Is that what your skin actually does? Does your skin actually float in space? The top layers kind of float, hovering over the rest of your skin?
No.
No. Remember, one of the things that we talked about is that there are artifacts that occur sometimes when we are processing tissue. Let's take it out of the slide and go into real life. You've got tomorrow off, so since you have tomorrow off, you're going to sit out in your backyard because you're not allowed to leave the house, laying naked on a towel, reading your textbook. And after doing that for about 10 hours, you might get a little bit of a sunburn. And as you get that sunburn and the skin repairs itself, those damaged cells need to be removed. When you lose your sunburned skin, does each individual cell just slough off by itself?
No.
No, it peels off in a layer. And the reason it peels off in a layer is because these cells not only have keratin in them, but as we talked about, they have a large number of desmosomes that lock them together. They also have this kind of sealant, almost like a wax, that fuses them together. And so what happens is, even in death, these cells stay stuck together. Now remember, with this skin, we are taking a chunk of skin we had to cut out, we had to freeze it, we had to use a really thin knife to cut it. And often when you're doing this with thin skin like this, the tissue layers will actually shred. Well, they'll actually separate from each other. So again, this is not what it looks like in the real body. This is just a case where we have that shredding of the tissues, that separation of the tissues from processing it. So it's that artifact that we're seeing here. So they don't really float in space above the other layers, but we can still see it pretty clearly what this is. All right? Also, because you're going to be working on it because it is due on Thursday, what I will point out to you is, you see this dark pigmented stuff down here in the deepest layers of the skin?
Yeah.
That's melatonin. This is the skin of a deeply pigmented individual. All right? Whereas over here, where you see very little, this is what the skin would look like in a very light-skinned individual. That's it. That is the biological difference between a black person and a white person. When you look on the news, when you look outside and see all the tremendous problems and unrest that are going on, and from a biological standpoint, when you see how incredibly meaningless these differences are, insignificant these differences are physiologically, biologically, it is astounding the issues that we've been having with this. And I hope it is something that can be resolved eventually. All right? Because that's it. That's the difference. That's the biology of it. It's ridiculous how silly that is. Now, that was what we would call thin skin, like on your forearm. Here is skin like on the heel of your foot or the palm of your hand. This is what we would call thick skin. Notice, and hold on, let me get my eraser so I can get rid of some of this stuff. Oops, I didn't mean to erase that. I want that back. Again, when we look way down here at the bottom, we see densely packed cells with nuclei coming up. But notice now we see a massive, massive layer of cells that are all dead. Notice all of these, not a nucleus to be seen anywhere. Hundreds of layers of these dead cells, which give you that very thick, coarse, calloused palm, that very thick, coarse, calloused heel of your foot that we see here. That's where you get that pumice stone and scrub off these rough layers of the top of this skin. So this is also a keratinized, oops, didn't mean to do that. This is also a keratinized stratified squamous epithelial tissue, but with a much thicker layer of dead cells on top.
Question.
Yes.
How can you distinguish if it's a thick layer or if it's a thin layer?
That's one of the things so the short answer to your question is that's what we're going to be doing on Thursday and Friday. On Thursday and Friday, we're going to actually identify all the sublayers of this, identify exactly what's going on in each layer, identify the layers, and we'll focus on telling the difference between thin and thick skin. So that is what we're going to be doing over the next couple days of class. So I don't want to focus on that right now, but we will get there.
Okay.
All right. Did I say melatonin, Ryan? I meant melanin. If I said melatonin, then I misspoke. I meant melanin. Melanin is the pigment. Melatonin is the stuff made by your pituitary gland that makes you sleepy. So I apologize if I misspoke. Sometimes I'm busy getting on my high horse. I don't think about what I'm saying. All right, excellent. One last tissue that we are responsible for here.
Can I ask a quick question about the 2 slides ago when you were saying the melanin difference? Is that also, because that's not going to be how you see it in the skin, right? That's also the pigmentation that we have put to see the
I'm
color?
sorry, this?
Yeah.
No, this is the pigment.
Okay.
It is stained to highlight the nuclei and things like that, but the melatonin pigment that stands out, the dark melatonin pigment, it doesn't pick up the stain the same way. So the color you're seeing there is pretty much the color of the pigment.
Okay. Got it.
Yeah. All right. Identify the tissue type. What's left?
Glandular.
Oh, okay. We haven't gotten there yet, but there's one before that one.
Transitional.
There you go, transitional. Excellent. Now, again, how do we know that? Because it's the next one on the list. Remember, as we've talked about, as we've seen, when you look at the basal surface of this, again, we see small cells packed tightly together. It all looks the same. But notice this time, what makes this tissue different from the stratified squamous is notice this time, as we move towards the apical layer, the cells actually get bigger, right? And our stratified squamous is the cells flatten and get smaller. But here, the cells are actually getting bigger. In fact, we have these big, huge block, oops, I didn't mean to do that. These big, huge block cells on the surface of it, right? So this difference in the transition is that as the cells get bigger is one of the things that we have this transitional epithelial tissue. Identify one of the locations where you'd find transitional epithelial tissue.
Bladder.
Bladder,
Ureter.
ureters, or
Lungs. Or
nope, urethra. Remember, the ureters are the two structures that carry urine from the kidney to the bladder. Then the bladder, and then the urethra is what carries the urine out of the bladder into the bushes behind your house, or the toilet, or wherever you go, right? Now, as I said, one of the ways to identify this is that the cells get bigger as they go towards the surface. And we have these big block cells on top. However, as I also mentioned, we're always looking for dead giveaways that are like, boom, instantly you know what you were looking at. And when you can get those things, it is going to make it much, much easier for you. Now, this doesn't happen with every cell in the transitional epithelial tissue, but I want you to notice something about this cell. What do you notice that's interesting or different or unique about that cell that we're pointing at right now?
is it the nucleus? It's not perfectly round. It's or no?
Well, close. It has something to do with the nucleus, but what do you notice about the nucleus of this cell?
There's two.
Yeah, it has more than one. It has two nuclei. If you see a big block apical cell on top that has two nuclei in it, that is a dead giveaway. You are looking at a transitional epithelial tissue. Transitional epithelial tissues are the only epithelial tissues that will have these double nuclei cell on the apical surface. Why it occurs occasionally is not understood, but it is something histologically that is consistently found. And so if you see it, you know it's a dead giveaway. You were looking at a transitional epithelial tissue. All right. Questions on that? All right, excellent. We only have, we're on time. We're doing good. One thing left. That one thing left is this right here. Now, remember, when we are looking at our exocrine glands, and more specifically our multicellular exocrine glands, there are two things that we are always looking for. The first are the ducts. Ducts are going to be made up typically of cuboidal, or in some cases columnar cells. And remember, those are the conduits that are carrying things out. And the other thing we are looking for are the secretory structures. Now, starting tomorrow, and I guess by tomorrow I mean Thursday, you are going to be responsible for learning glands of the skin. But for right now, there's only one gland you are responsible for. And that one gland that you are responsible for is the salivary gland. And remember, as I mentioned, salivary glands are responsible for producing a very complicated substance known as saliva. All right? And as we talked about, saliva has two parts. It has the watery part, and it has the viscous, sticky part, the mucus part. Now, as I happen to look at these cells right here, like those cells right there, what do you notice about the color of these cells?
They're light.
Very
don't have a
light,
color.
very clear, very kind of pillowy. Remember, one of the things we talked about is mucin. The protein mucin typically doesn't bind to stains. So cells that have a lot of mucin in them are going to be much lighter in color. So these are the cells that produce the mucin part of our saliva. Okay? Now, let's look at the shapes of them. Notice as we look at the shape of them, and I'm going to use a highlighter to work around some of these. Notice this one is kind of circular in shape, right? But notice this one's much longer in shape and kind of has this weird bumps and stuff to it, right? Think of it this way. If I have a ball of twine, and I cut that ball of twine in half, are you going to see the single strand of twine that's making it just wrap around and around and around again in that section that you cut through it?
No.
No, you're going to see a bunch of cut ends at all sorts of weird angles. And that's what we have here. When we have these weird angles, what we actually have is a coiled tube. So what these are actually mucus tubules. These are tubules that are responsible for producing mucin. All right. Notice though, and I'll switch my highlighter color. We'll use green this time. I also have these secretory structures that are much darker in color. And that one's ball shape, and this 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 one's ball shape. Notice every single one of these things is a dark, compact, ball-shaped secretory structure. Well, since these are all ball-shaped secretory structures, they're, of course, alveoli. And they're not light in color. So they're not making mucin. So what do these alveoli make? Well, they make the serous fluid. So we would refer to these as serous alveoli. Oops, so I spelled serous correctly. These are serous alveoli. Their job is to make serous fluid. All right, and then these things together, the serous fluid and the mucus, make our saliva. So what we have here is a salivary gland. We can recognize what are really the three parts of this salivary gland: the duct, the serous alveoli, and the mucus tubules. And because this is a gland, the other piece of information we're responsible for is its functional classification, its structural classification, and the substance it produces. Let's start easy. What is the substance that is produced by this particular gland?
Mucus.
True, but we could even be more specific.
Saliva.
Saliva, there you go, excellent. What this produces is saliva. Excellent. What is the functional classification of a salivary gland? What did we say the most common one was? I know we just did it like an hour ago, so you're really having a chance to process it, but I'll wait while you look back at your notes. What is the functional classification of a salivary gland? Merocrine, excellent. Perfect. And lastly, what is the structural classification of our salivary gland?
Mucus tubule.
Well, but remember, it has mucus tubules, but it also has serous acini. And even though we only see one duct, if it's going to have two different types of secretory structures, it probably has two or more ducts. It's got definitely two or more secretory structures. Bingo. So structurally, this is a compound tubulo-alveolar gland. So there you go. Based on the stuff we talked about in lecture, based on what we're looking at here histologically, we should be able to distinguish all of these parts. Again, I do not expect you to have mastered the histology after doing this. This was us quickly going through it so you understand what is expected of you. Your job now is to go through, fill out this handout, and make sure that you have mastered this information. All right? Questions on that?
Will there be more exercises related to this on our study lab in my lab?
So yes, if you go to your Practice Anatomy Lab, there is histology on there. Then obviously your textbook, your lab manual have those stuff in there. I don't think there's an interactive physiology for tissues. There may be for glands, but no. But that's why, again, that's why you need your histology atlas. That's why you need those other resources so that you can have good examples. Because the histology atlas doesn't just do a good job. Where's my histology atlas? I've got a couple, but I want specifically the LaBeouf one. Here we go. Your histology atlas doesn't just have great pictures, but the other thing that they have is great descriptions that describe what it is that you're looking at as you're doing these things. At the beginning of the chapters, there's lots of words where you can read to help to make sense and understand this. And yes, remember, not only to answer the question from before, hold on. Anatomy lab, yeah, the practice anatomy lab is under the study tools in the Master in A&P, yes. And yes, Michelle, this is not only your study guide, but I expect you to fill it out and also label them. So if we, again, it doesn't have to be the most beautiful artistic thing. Let me totally cheat and just do this real quickly. Hopefully you spend a little bit more time on this than I am doing. But what you're gonna do, so for instance, for our, hold on, I gotta move this out of the way so I can see what I'm doing. There we go. So if you notice right there, I drew my renal corpuscle, right? But remember, every single thing that is on this list, you need to have labeled over here. I don't want you drawing lines to these things over here. I actually want a label coming off of it, where you have a label coming off of it and it says, Bowman's capsule, right? So that you have a label coming off of it. Because what I want this to be is I want this to be a study guide where you don't have to use the words, where you can just look at the picture and you can be able to identify and see all the things. So everything that is on the list over here should be drawn here and should be labeled here. And like I said, you don't have to use color. Color's nice. It's fun, makes it more entertaining, but it's not required, but you can do that. And yes, to switch to here. Again, I told you guys that you should have print these out ahead of time, but remember, come to your modules, under the section, this is where you're gonna find all the information. This is where we spend most of our time, is here on the modules. Here's where you turn in your assignments. Here is where the lecture slides are located, and here are the lab handouts. So notice there's an epithelial tissue handout and a connective tissue handout. These are the two handouts that you need to print out. You need to do the drawings, and now we've done everything for the epithelial tissue. So now you've got that to be able to go through here, right? This is also where the lab simulators are, your answer keys are, and the pictures that I've been taking from the lecture slides. So all of the stuff, most of the stuff that you need for the class is here under the modules. And then the Master in A&P will be how you get to the study guides and things along those lines. No, I do not want the pictures printed out. I want you drawing them. Learning is tactile. I don't want you to just cut out pictures and label pictures. Again, you've seen my drawing both during lecture and on that. You're not being graded on your drawing skills, but I want you to have that tactile practice of drawing it, because then what that allows you to do is on your drawing, like I did. Remember, when we looked at the kidney, there was a whole lot of other stuff going on as well. But what I'm able to do with my drawing is just focus on the renal corpuscle, just focus on the parts that I'm responsible for. And it's going to allow you to be more specific and more precise to know what it is that you're looking for. So it is important to do those drawings yourself. Again, you're not going to get graded on the drawings. You are getting graded solely on doing the activity, filling them out, and labeling them correctly. Make sure you put everything that's there is you label. I don't want numbers. I don't want lines going to the activities. I want you physically writing out the pictures, physically writing out the labels. And remember, that's due on Friday. So then you're going to have that done on Friday, turn it in for your 10 points from me. But then on top of that, you're going to have that great study tool to help you to use it for the lab exam so that you're ready for Monday. So it's a win-win all the way around. You get free points for doing it, and it's going to help you to study and master the exam. All righty. Awesome. Any other questions on any of that?
I had a question. So couple questions actually. When we attempt to resubmit assignments, you have all the copies, or does the last copy only stay with you?
So when you submit it, I get all of the copies. So so for like the Physio X's,
Yeah.
I know it's scary because it says resubmit and it only shows your recent one, but I'm able to go back. So if you do your Physio X's and you do 3 today and 2 tomorrow and 1 the next day, and you turn them in on different days, I am able to view all of them. I see everything that you submit. So you don't have to worry about that. You're not, if you resubmit something, you're not erasing what was there before.
Gotcha. And for Physio Labs, you said that's also where you, is it graded per your performance, or that you finished it? Or?
No, I'm grading you for completeness. Again, if you only get 75% on the post quiz, you're not going to lose points for that. However, if you don't answer 2 of the post quiz questions, then yes, then you will lose points for that because you didn't do the work. Right? If you guessed wrong with a hypothesis, or if your answer to a question is incorrect, I'm not going to take away points because, again, the point of this is to be a learning experience. If you don't answer the review question, or if the review question says, do you think something's going to be A or B, explain your answer, and you just write A, then that's not putting time and effort. So I'm looking for time and effort on those.
And sorry, one more question about that. If we are totally missing the mark on one of the questions that are the essay questions, because there are free writing questions too in there, will you be, do we need to send you those just to get a better idea of whether we were right or not, because it doesn't grade your essay questions, or will you be able to tell us if we were totally off there? Should we just like approach you separately, or?
Yeah, I think if you're concerned about a particular question, I think the best thing is to address me directly for it. Because again, you got to remember, with 5 of these from every person, and each of them having 2 to 4 review questions and 30 people in the class, that's a lot for me. So again, I am spot checking things and I'm checking them to make sure that they've been fully cleared out, but I'm not reading every word of your answer necessarily. I'll do that with 1 or 2 of your answers to see that you're getting the concepts. But that's also why I post the answer keys for the reviews, is so that you can see what the correct answer was supposed to be. They haven't given us those for the Physio X's. So if there is a concept that you're not clear on, then I would encourage you to ask me directly about it, and we can discuss it that way.
All right, thank you.
Excellent, great questions. Any others? All right, go ahead. Michelle, did you have a question?
Oh, no, I'm good.
Excellent. Well, we have, like I said, we still have 35 minutes, 40 minutes left of class, but I'm not sure that's enough time to get all the way through our connective tissues. And rather than starting it and getting halfway through, I think we're still doing okay time-wise, where our best bet at this point is to go ahead and call it for the day today. We have a lot to cover then on Thursday and Friday. We need to finish connective tissues, then we're going to put those connective tissues together to make membranes. Then we have the anatomy of skin and the physiology of skin. So we've got a busy couple days ahead of us left, a ton of material still to cover. So like I've said before, when you have a day off like this, not only should you be looking at the material we have already covered, but you should be looking ahead as well, preparing for the next class. So look at connective tissues, look at membranes, look at skin, because I don't want that stuff to be completely fresh when we hit it on Thursday. Well, I want to hit the ground running and make sure we understand this material. All righty. Any other questions? All right, excellent. Well, in that case, have a good day. Be healthy, be safe, take care of yourselves, and I will see you. Enjoy your day off. I will see you Thursday. And like I said, if you haven't done so, I encourage you to meet with Jeff. Go to the Open Lab if you're able to. I think Jeff is an incredible resource. He is an amazing instructor. And the fact that he is still able to work with the students, I think, is excellent. So I strongly encourage you to take advantage of him. All righty.
I apologize. Where is it under again, the Open Lab?
So the instructions for them are under the study tools and the modules. If you go to the modules, the instructions are there, and actually the link to it is there as well. So there's a link there to he's got a static Zoom address that he opens up on Wednesday. So those instructions, there's 2 places to get it. If you look under the pages, there's a page on the Open Lab there, but the link to that page is under the modules, under the study tools.
Got it.
All righty. Any other questions? All right, you guys, have a great day.
You too.
You too. Thank you.